We're on? Okay, so good morning, everyone. We are a few minutes late, thanks to subway problems and so on, but we're ready to start. And so I already introduced Christoph, so no need for more introductions. He gave us a really nice talk yesterday about the historic perspective of synchrotrons, and we learned a lot. And today he'll tell us about metal speciation with X-ray fluorescence imaging. So thank you, Christoph.
Thank you. Thank you, Liliana. I can tell you by tomorrow morning, you'll be completely tired of me. Yeah. Okay. Well, wait. So what I will what I will show you today is few examples, actually scattered examples of what we've been done with imaging using synchrotron radiation. At different scales and why we've done that, and also how we did it. And I won't go into the detail of the research completely because I'll be doing that tomorrow, actually. So if you have any additional question, we can discuss later. But okay, so the purpose of the talk is really to give you an introduction to imaging with photons, and also I hope give you the idea to do some some imaging techniques and how is it useful and etc. From my point of view, actually there is the window of the Zoom that is yeah, I can. Yeah, that would be better like that. Okay. From my point of view, the problem of imaging and speciation at the same time is a problem of scale. You know, it's kind of obvious to say that imaging is a problem of scale. But because when you get an image, immediately you think about what is the scale. Okay, you can use optical, so that's photons, but in the visible range, and then you are in millimeters or micrometer scales, but for sure not angstroms. That's not possible. You can use electrons, and you do electronic microscopy, and that's very useful as well. And you can use photons, but in the X-ray regime. I am doing environmental chemistry, and for environmental chemistry, we try to understand the speciation, and this is also a problem of scale. I like to start from two extreme cases. This is a map of a site in the US in Colorado, and it's a former plant, whatever it is. It's not of interest here, but it's a former plant of few kilometers wide. So this picture here is more than 10 kilometers. Okay, and when you're studying such a site to try to implement remediation techniques and understand why it's polluted and how it's polluted, etc. The real answer, the real picture you want to have is this one. You want to see a molecule, okay? Because you want to understand how does this molecule interacts with the environment and how it gets contaminated and messy and all that stuff. Okay, but so you start with this question, and you want to end up with this answer. Okay, and that's how you understand how to go from here to here. Okay, and as I pointed out, that's a factor of 10 to the minus 13. So it's completely enormous. Okay, and it's actually not feasible. So it's kind of a challenge. And so when you are speaking about speciation, so I recall the definition of speciation here, but so when you're speaking about speciation immediately in the environment, you need to speak about the scale. What is the scale? Okay, because it can be so heterogeneous as well. Okay, and then I like to quote this sentence from is a French science philosopher that you may know, and I think I like the sentence because it's a little bit provocative. It was from the first part of the century, I mean the 19th century, and he developed a lot of philosophy about rationalization in science. And so basically what he says, it would be better to be completely ignorant than have knowledge deprived of its fundamental principles, which of course it's a little bit extreme, but I like it because we should never stop on our way in understanding things. We should always try to understand up to the maximum point where we can understand things. And that goes back to my picture here. When you have questions about remediation of such a size of 100 kilometers square, you should go up to this picture here and really understand what's going on with the chemistry of the contaminants there in order to answer this question. And actually, I work on that side with my American colleagues. My part was very minor, but I really like this example because you know, this was a really tough military plant in the US, and they cleaned it up. They completely cleaned it up, and now it's a field with absolutely nothing on it. So I think it's quite a challenge. And how did they do that? They did that at the synchrotron, not doing imaging because at that time it was not possible, but we did that at Stanford actually, by understanding the speciation of the elements that was plutonium in there everywhere. So the speciation of plutonium inside the walls of the, you know, and on the grass and on the soil and everything. And by understanding the speciation of plutonium in this factory here, so I recall it's 100 kilometers square meter, they could clean it, you know. And what's completely amazing, they clean it with trucks. You know, they brought trucks and they just removed everything. But because we understood the speciation, before that they couldn't do it. Okay. So I try to put a general picture of what is the scale. Is it what is I mean, what is the scale in terms? Is it meaningful? What is a meaningful scale when you're speaking about speciation? And when you deal with environmental samples, but not only that, when you deal with cells or living species, you deal with complexity, mixing of species. And that's the hardest thing to deal with. You have a mix of species, a lot of species. And so if you have what I call speciation complexity here with a special scale in here, of course you have the molecules here, so that's the easy part, okay? And then you have the proteins here, okay? I put colloids, particles, of course, that's a few examples. Sorption processes. I put the living cells here because it's small, but it's very complex. You have thousands of chemical reactions in living cells. Aquatic chemistry, on the other hand, is very homogeneous by definition, okay? So it might be a large scale, thousands of kilometers, but it's still the same speciation because it's average in solution. On the other hand, geochemical samples are probably the most difficult to understand because it's very heterogeneous. Okay, you can sample at one place, and the next meter you will get completely different samples. Each time I get two. Okay, that doesn't matter. Okay, so I think it was useful to recall what you've seen yesterday, but with a different picture. I mean, it's the same picture, but in a different way, but it would be short. But anyway, I always thought as a student, it was useful to hear the same thing with two different people. And so what we are dealing with is a core electron spectroscopy, and that means that we have, I use the 1S because it's the most common, but as you've seen yesterday, you can use the 2S, the 2P, etc., etc. So you excite an electron here, which goes in the atomic potential in here in the valence states, or higher than the valence states actually, and creates a hole here. And that's the absorption phenomenon. So as soon as you do that, then you have an absorption band in here. That's a photoelectric effect. Okay, so that's really the what Thomas said. It's the real the direct absorption, the real phenomenon. Okay, so that's what you get in here. But you can have also relaxation, or it's not you can have your relaxation. You will have relaxation, and you're free to watch it or not. And so when the atom relax, you have a transition from an upper level to the core state, and you have emission of a fluorescence line. Okay. And I was very glad that you've seen that yesterday. Actually, it's not the same age, but it's the same idea. I think it's really nice to see what the spin-orbit coupling is, because we always talk about the spin-orbit coupling, but it's completely phantomatic. But here you can really see it, as you've seen yesterday. You can really see the splitting between the G3.5 and 5.5, because this splitting is pretty high, because you are dealing with core electrons in here, so they are very deep in the atom. So this splitting here is very, very high. So I won't go into the details because you heard that yesterday, but you can also calculate the intensity, etc., and etc. So we want to do imaging using this phenomenon. Okay, so we want to couple imaging with spectroscopy, and that's the main point. So you could do, so how do you do imaging? Okay, you take light and you look at your sample. So there are two ways of doing that. You look through your samples, okay? That's the easiest way, but it doesn't work if your sample is occludes your photons, okay? If your sample with visible light, if your sample is dark, you're lost, okay? The other way of doing, and you can do that with electrons as well, okay, transmission electron microscopy, okay. But you could look indirectly at your photons or your electrons, that's SEM, or you can, or fluorescence with a photon. So actually what you would be looking at is you excite these core electrons in here, and then you get fluorescence lines in here. You may choose, as we've seen yesterday, many fluorescence lines, and then you end up with a Rix map, as you've seen. But you may select one fluorescence line and focus on one line and get your absorption spectrum from this line, as you can show as you can see in here. Okay, so in the examples I'll show you, we've selected one line. So just to make sure you really understand that, because that's very important to understand X-ray absorption spectra. When you look at it in the fluorescence mode, let's say you look at this emission line in here, and what you do is you look at the modulation of this line as a function of the incident energy. Okay, that's really what you're doing. Okay, so you focus, you gate this line in here, and the modulation of the intensity of this line with the incidence energy will reconstruct your absorption spectrum. Okay, that's what you're doing when you are measuring in fluorescence mode. Okay, I just wanted to make sure it's clear for everybody. Okay. Okay, so in summary, we have a field or a piece of sample. We want to look at it. We want to see what's going on in the sample at different scales, and we want to do a spectroscopy exactly on the same sample to try to understand the speciation. The speciation is here, and the image is here. Okay, we need both. Okay, we really want both. It would be easier to get only this one or this one, but that's not what we want. Personally, I use two different setups of beamlines. So the first one is the beamline we use in Berkeley. It's ALS. It's called the Stickson beamline of scanning transmission X-ray spectroscopy. Really, what it is a microscope. It's a microscope, but with X-ray photons. And so, as I told you yesterday, it's the same image. I mean, it's a very simple experiment, but you do it with X-rays. And of course, that makes things very complicated. So in Berkeley, there is a Stickson beamline where, so you have the storage ring, and basically how it works. So you have a Fresnel zone plate. So at this energy, you cannot use mirrors anymore. You have to use zone plates to select the energy and to focus the beam. You have an order sorting aperture plate. It's like a pinhole just to select the right harmonics that you want. And then you have your samples in there, and then you detect the beams going through your samples with X-ray. So it's very simple. Okay, you could do it with fluorescence detection as well. You could put a fluorescent detector in here and look indirectly to the fluorescence. Okay, we haven't done that, but you could do it. Okay, and so that's what you've been doing at the Hermes beamline as well. It's exactly the same setup. Okay, so that's a Stickson beamline. In Soleil, we do other type of imaging. We use KB, so it's a Kirkpatrick-based mirror setup. Basically, what it is two mirrors that are placed orthogonally from each other. So one is like that, and one is like that. Okay. So the beam, it's a top view and side view. So the side view, the beam is coming this way. Okay, is going to the first KB here, to the second KB here, and then to the the first KB here, second KB here, and then the sample. So the KB is a bent mirror. I mean, two mirrors like that, and it will focus your beam on your sample. Okay. The advantage of a KB mirror is you can use it at high energy. You cannot do that with a zone plate. You have to be at low energy. So basically, with a Stickson beamline, you are stuck at the lowest energies, while with a KB mirror, you can go to the higher energies. Okay, so depending on the beamline, for nanoscopium, for instance, they were going to mid energies. On the Mars beamline, we can go to very high energies because that's what we want. But the drawback on that is we have a less resolution, spatial resolution. So just to give you, yeah, a few hundred to maximum. I mean, Berkeley, I think this one is up to 2,000 or 1,000 electron volts. And here on Mars, higher energy, we can go to 20,000 electron volts. Yeah. So you basically here you're stuck. Well, I mean, you're stuck with you're dealing with NEXAFs. That means oxygen KH, carbon KH. I will show you an example of lantern eye MH. So below 1,000 or maximum 2,000 electron volts, and here higher than 10 kilos, basically. Okay. But nanoscopium is in between as well. Okay, that's a few pictures of the Mars beamline where I used to work. So the KB mirrors are in this box in here. The beam is coming from the right, and we put the sample in here, and we can put the sample in the cryostat. Okay, so if it's, of course, it's much easier to work at higher energy because you don't have to have all those vacuum pumps and etc. and you can, but you're not looking at the same thing. That's the point. Okay, so we have our sample in here. We have the fluorescence detector in here, and the beam is coming this way in here. Okay. So I divided my talk with 3 scales, typical scales, to try to show you what we can do. So the first scale is the largest one. I call it geochemistry scale because we're looking at large samples. Okay. The idea was to, we wanted to study the speciation of, it was uranium in a column of soil. So it's a specific soil, and it's a specific source term of uranium. I probably don't need to detail this, but we use 2 types of uranium source, hydroxide and an uranium metal. And then it's like if we were reproducing the environmental conditions. So we those columns are, you know, middle size, a few tenths of centimeters high. And so we have our soil in here. We put our source in here. The source is here, and we weather the column as well, as if it were in the real environment. So we have water flushing from here. Okay, and then what we do is we cut the column like that. And we cut the sample. We go to get to the part where the source term, the source is, and that's a picture in here. So we cut everything in slices. We use resins and etc. It's pretty complicated, but anyway, we end up with a sample that's few centimeters. Okay, maybe 2 centimeters wide and maybe 10 centimeters high. Something like that. First of all, this is because I think the picture is very nice, but there is no real result from this picture. This is an optical picture. Okay, so you see it's millimeters in here, and here is a piece of metallic uranium. Okay, and we've been weathering this for months. Okay, and so the question was, can we start seeing any alteration of the uranium here? And is there a start of migration of the uranium from here? Oh, the substrate is calcite. Okay. And so here you have the XRF map. So to do the XRF map, we are after the uranium age, and we are measuring, can come back to, yeah, this, and we are measuring, for instance, it doesn't matter, well, this emission line in here. Okay, that's how we do it. Okay, so in here. Okay. So that's a picture of so it was a very thin piece of uranium metal. Okay, but that's okay. That's the first step. But we want to see the speciation. Okay, so what we did is we scan vertically like that and acquire the Xenon spectra of the uranium. So the Xeons are very good when you are on the metal, of course, but they get very noisy when you are below the metals. And why is that? It's because there is no migration, okay, or very little. And so you see the Xeons, the picture is not very clear, but there was a XANES here just below the metal, and that's this XANES here. So there is some uranium, but it's just too low. You know, we can't tell what it is exactly, but there is some, but it's very, very small. Okay, so by doing this, you can see the evolution of the plum, okay, below the metal, and you can try to have a rough idea about the speciation of inside this plum. Okay, so that was the example that didn't work. So now we start from uranium hydroxides, and each time you have the optical image on the left and the XRF image on the right. Okay, this is pure calcite with water. This is pure calcite with humic acid substances that you find in the environment. Okay, or organic substances. And immediately you see that you have different information with the optical image and with the X-ray image. You know, with the optical image, you see a dark line around the uranium source. Okay, and with the X-ray image, what you see is a plum here of uranium. Okay, so you have migration of uranium countercurrent, because the water is flowing this way. Okay, so you have migration to the top, okay, which is very strange. All right. Okay, and that's a natural substrate. So that's pure calcite, and that's a natural soil. Okay, so same picture in here, and about the same in here. And again, you have migration. It's not to the top; it's to the left a little bit, but it's not to the bottom. Okay. Yes. No, the square is a real sample. Okay, I haven't been clear enough. The square is here. It's a source. It's our source. It's the real source. It's the source we made is 2 millimeters, 5 millimeters. So the source is a rectangle. Okay.
Okay, thank you.
So what you see here is a cut of this source. Okay. So this size here is the size of the cut of the slice of the source, actually.
Thank you.
Okay. So that's the pictures. And so what we did is, well, of course, we did some EXAFs on different points and on different lines. So above, on the dark side, here in the middle, but we know what it is in the middle, and below. And well, the first point is that we have migration now. Okay, we have migration below the source term because we can acquire the EXAF spectrum. Okay. Well, to make a long story short, actually, what we have is a migration of the uranium here and a slow conversion of the speciation into uranium carbonates because it's calcite. Okay, but with different steps. It's a little bit more complicated than that. But that's not the point here. The point here was just to show you that once you get the image, you can acquire the EXAF spectra in every point, wherever you want, where it takes time. And then you can try to work out the speciation. Okay, this is more interesting and complex because now it's a natural soil. So we took the soil from the field. We took a source that we made, and we reconstruct the column as if it was in the field. Okay, so everything there is natural except the source that we made, and then we weather it the same the same way for months and months. Okay, and now you see something completely different. Okay, you have the source in here. That's a source, but part of it is black and part of it is yellow. That's the optical image, okay, and that's the X-ray image, okay. And clearly, what you see now is a ring around the source, okay. Well, there are two things that are very surprising here. Is, well, you have an isotropic distribution of uranium around the source. That's a first. Sorry.
A horizontal or vertical cut?
It's vertical. I mean, it's a vertical cut. Each time it's a vertical cut. Yeah, I probably haven't been clear at all.
Okay. So,
yeah. No, it's always vertical cuts. Yeah, the cut is this in this direction here. So you see, this is in the column will be like that, okay, and that's a cut through this slice here. It's a vertical cut. See, so water is going from top to bottom. Yeah. Okay. So you have an isotropic diffusion of the uranium around the source, and you have a depletion zone in here. It's as if the uranium was migrating away from the source, but just around the source, you don't have any uranium anymore. And that's very, very surprising. Actually, we still don't understand. Okay, and so we did xanes on all those points, okay. And what we've seen is a reduction of the uranium going from the center of the source to the bottom in here. We have reduction of uranium-6 into uranium-4, which is really very strange. So that's a real nice example because you have a real environmental complexity in here. You have oxidoredox processes going on that we still don't understand. And you have, again, you have isotropic migration, although the water is coming from the top to the bottom. Okay. So all of this is, of course, very, very interesting. Okay, let's move to smaller samples, living species, but still few millimeters. So it depends who you are talking to, but for some people, millimeters is just enormous. And if you are talking to geochemists, you know, kilometers for them is just nothing. And so but that's, I will be talking tomorrow. We were looking at the contamination of sea urchins in living sea urchins in aquaria. Still with uranium. We like uranium very much. And this is a picture. So I won't get into the detail, of course, of the data, but just show you the picture. Here is again XRF map of one spine of the sea urchin. Okay, and the question here was to understand if there were some uranium hotspots, precipitation, because a spine of sea urchin is magnesium carbonate, sorry, essentially magnesium carbonate and calcium carbonate. So it's carbonates, and uranium likes carbonates a lot. And so the question was to understand if there would be some hotspots of uranium on the surface of the spine. Okay, you could imagine that uranium would sorb on the surface and then sorb as a carbonate complex on the surface of the spine. Okay. And so we did the X-ray map below, and of course after the uranium age. Okay. And what we see is just a difference of color, but that doesn't mean anything because that's that's how you just scroll the color bar or the sensitivity of your image and you change the color. So it doesn't mean anything. What it means is that you don't have any hotspots. You don't see any hotspots. You see a global change of color, but you don't see clearly hotspots of uranium on there. So what it means is actually, we know that uranium is sorbs on the spine, but like a monolayer, something like that. And so we did some xanes of that. So the xanes is very, very difficult because it's only a few ppms of uranium on the spine. And so the xanes is the dots here. And we can look at the derivative and try to understand the speciation. Anyway, we came up with a speciation of uranium on the spine, but the question we answered is there is no hotspot. It's very heterogeneous. Okay, then we looked at the gonads of our sea urchins. And again, I'll give you the details tomorrow, but what is more interesting is this image in here. So that's a stick-sum image. So that has been done in Berkeley. This is the N4 edge of uranium. So it's very low energy. It's 738 electron volts. And so, so we are looking in transmission. So it's transmission microscope, okay? And so you see the cell wall in here, okay, very clearly. It's very nice, actually. It's amazingly nice, actually. When we saw it, we were so amazed. And you see the organelles inside the cell. It's very clear, okay. And so then we did that above and below the edge of uranium, and then we subtracted those two images. And so you know, so we did a contrast image, and that's the image we got, okay. Well, first glance, you say nothing, okay. We don't see anything, but we know there is uranium in there. We know it because we did some ICP-MS. So we know there is a lot of uranium, and this is an exact spectrum of the gonads in here. So there is a lot of uranium. So where is uranium? Well, if you look very carefully at it, what you will see is a ring in here, okay? You do see the ring, okay? But it's very shallow, okay? And why is that? It's because the uranium is actually on the cell surface, on the cell wall, but it's very well distributed on the cell wall. It's, again, it's not precipitates of uranium. It's not hot spots. It's very homogeneously distributed. Actually, it's coordinated to a protein. And so I'll tell you yesterday, but we found the protein, and so that's for you. And so we describe the protein and the complex inside the protein, etc. But that's also the drawback of the looking in transmission. Yes.
So the first image, it's like a natural like image of the cell, and the other one is from the X-rays.
No, both images are from X-rays. Okay. But that's at the fixed energy, okay? And this one is a result of the subtraction of this image below the uranium age and after the uranium age, okay? So that's a subtracted image. That's a difference of two images, okay? Below the edge, you shouldn't see the uranium. After the edge, you see the uranium. So the difference shows you only where the uranium is, okay? If you make the difference, I mean, very, gentle. I mean, that the difference is tricky to do. Okay, I didn't do it myself. So they did it in Berkeley. Yeah, but no, it's difficult to do. But anyway, it's because we are looking in transmission. We are not as sensitive as if we were doing the measurements in fluorescence. So probably it would be better to do these measurements still with a stick-sum, but in fluorescence mode. Okay, but at that time they didn't have the setup. Okay. So the answer is uranium is on the cell wall and it's coordinated to a protein. Okay, another example with a muscle. Same question. This is an optical image. So this is a digestive gland of a muscle. Okay, that's 1 millimeter. So it's a muscle. It's an average size muscle. You know the size of the muscle, so again, we put on the optical image, we put the X-ray image, so on top of it, same scale, of course, and you see a diffuse contamination of uranium in here. Okay, and when you go out of the digestive glands, you don't have any uranium anymore. So we did exams where it's pretty noisy because it's very diffuse, but believe me, we know it's uranium-6. And here it's the same with the byssus. I don't know, I'm sure you know what the byssus is, but you might not know the name. The byssus is the filament that comes out of the muscle. The muscle uses the byssus to stick on the rock. Okay, so that's obviously something that you remove when you eat the muscle. Okay, and it's a very strong filament actually. And so you have the byssus in here, and you can clearly see that the byssus is full of uranium. Okay, so the filaments are very tiny. Okay, and everywhere there is a filament, there is uranium. So there is uranium everywhere on the filament. Okay, so we took some XANES image on different spots, and actually the XANES are not completely the same, which means that there is a difference of speciation depending where you look at on the filament. So we've done some TEM images and other. We are doing some SIMS as well, imaging, and we think we have an idea of what's going on. But here, what you see is a difference of XANES. You see this XANE is different from this one or this one, and this one and this one are clearly different. Okay. But we need other techniques to complement the X-ray technique in order to understand what's going on. No, less. Yes.
Okay.
Yeah.
And about the
Well, I would say less, but it depends on the energy. Yeah, but usually less.
and about the diameter of the beam, this photograph that you show us of Berkeley, it was
This,
a nano beam,
yes. Oh, sorry.
and the diameter of the beam that you use at the soil was millimeter.
No, here the beam is about 40 nanometer square. Okay, in Berkeley. For the soil images, the beam was around 40 micrometer. So it's from nanometers to micrometers.
You use some other special optics to reduce the diameter of the beam?
Only the KBs. Here at Soleil, for 40 meters, for 40 micrometers, only the KBs, the KB mirror. We could use a pinhole. I mean, a pinhole, you could get down at this energy to even below 10 micrometer. Okay, but the drawback of the pinhole is that you lose a lot of flux. You end up with a very small beam, but with no photons.
Thank you.
So depending on the question, you have to really play with it and try different things. Of course, it takes a lot of time. Okay. Last example from Berkeley, actually. That was a very funny thing that we observed. We were working on contamination of sponges, marine sponges, and a marine sponge is it's very simple. it has a flux of water from outside and it has a tube inside. It's an osculum. And so the water flows this way and goes out this way. So this is the size of the cut of the sponge. So you see the osculum in here, okay, very tiny, and this is the outside of the sponge, okay. So that's a centimeter in here. Okay, so we combine TEM images, transmission electronic microscopy, and stick-sum images, and this has been done in Berkeley as well. And we look at different regions, so the close to the osculum and then on the outside. Okay, and again, you can see the cells. So there are different types of cells in a sponge. That's the spheral cells, and we can clearly see the cells. Okay, and that's 10 micrometers. Okay. But then we looked carefully at this image here, and we saw not very clearly, although we saw dots like that, small dots, very small dots. And when we looked at the TEM images, we could see those dots very well, like here, and dark dots. And that's not useful. That's not very common for a sponge. It was contaminated with europium, actually. So we thought maybe this is related to europium contamination. So, and that's really the nicest image I ever got. So we focused on one of those dots with a stick-sum microscope, and we did exams exactly on this spot in here. And this is an exact spectrum at the M edge of europium inside in this dot in here. So we could compare this exact spectrum with standards. Okay, and just to go through to the answer, we could understand that this particle here is a particle. It's a precipitate of europium carbonate, and that makes sense because then we looked at another region in here. And you see the spheral cells, okay, same picture, but you see a lot of dots everywhere. You have them everywhere, okay? So what's happening actually? And that we understood at the end. Well, europium has a very low solubility limit in seawater, but it forms europium carbonates. And we were below, I mean, we were below the precipitation of europium. So we had no precipitation of europium in our system. It was perfectly soluble. But the sponge is flushing water from here to the osculum and then out like that. As soon as the europium comes in the sponge tissues, there's probably a concentration effect, very small, but an increase of concentration, and it precipitates right away as nano particles of europium carbonates. And this is why we see much more around here than here. Here you have only a few dots, okay? And here you have a lot of them, okay? Because europium flows this way, it precipitates on the outside of the sponge, okay, and then it goes inside and goes out through the osculum. But by the time actually the sponge is filtering the europium and precipitates it in the same time, and that's how we see those dots. Okay. Okay. So to summarize. I would like to say that is speciation for environmental studies a question of scale? The answer is yes. Speciation is really a matter of scale. And what you may think as what is a speciation at a given scale might not be true at another scale. And that's, of course, very complicated. So to answer those type of questions, we have to combine several imaging techniques. Optics is still very good, and you can do optical microscopy, of course, as well. Scanning electron microscopy with EDX as well. We can do TEM as well, SIMS as well, and XRF. The real advantage of the synchrotron is that you can do the imaging and the speciation at the same time. That's very difficult with the other techniques. You might end up with a speciation. EDX, it will give you not fully the speciation, but the composition, elemental composition of your sample. But it's indirect. Only the synchrotron gives you a direct picture of your speciation. And of course, you can do X-ray fluorescence images, and you can do X-ray transmission images as well. So you have a completely large range of scales that you can study. And never forget the traditional spectroscopic techniques, because that always helps. You can do infrared imaging as well, Raman imaging, etc., and etc. So it's really by combining all those techniques that you can end up with a better answer of what is the speciation going on in your sample. I think I am done. Yeah, I just wanted to mention the beamlines where we worked. So ALS synchrotron, that's 1102 beamline, 1102 beamline, and the Mars beamline when we've done all those images. Thank you.
Good questions?
Thank you. I have a question about the intensity of the beam. Is necessary to change for each of these images the intensity of the X, right?
No, the intensity, the flux is fixed. The flux is fixed by the beamline setup. So what you do actually is you have your beam like that, and so you scan your sample like that, okay? And well, you can do two ways. You can have a single energy, and you scan your sample, okay? And then you'll get a one image at one energy, all right? You could do that and change the energy, okay? So you could do one scan at one energy, you change the energy, you do one scan, etc., etc. And there you have as many images as energy step, okay? That's called a stack image. And that's very interesting because if you do a cut through those images, you get a spectrum, okay? And that's exactly what we've been doing here. This spectrum here has been obtained by, this image at each step, at each energy step. So you obtain this Xen spectrum by cutting 100 images or thousands images at different energy steps. But it takes time, of course, you can imagine. So before you do that, you want to be sure that you want the spectrum.
And how long time takes for obtaining?
So a stack image here at Berkeley takes few hours. Let's say two hours or 3 hours. It's okay. A stack at Soleil because it's higher energy, takes much more long time, like 4 or 5 hours might take. Yeah.
Okay, thank you. Thank you.
And you probably mentioned that I missed it. This is a nanoscopy?
No, this is in
here.
Berkeley.
There.
This is in Berkeley. So it's the same, it's almost the same beamline as Hermes.
Here.
Nanoscopium would be comparable to Mars, but more optimized for imaging than Mars because it goes to a much lower, a much better spatial resolution. But it doesn't get to that high in energy. You always have to compromise. Yeah, you cannot get everything at the same time.
You cannot have it all.
Yeah.
Any more questions?
Thank you for the talk. And I found a particular interest about the example with the sea urchin. So I don't know if you have the citation of a paper about this.
I will talk about it tomorrow.
Okay. Thank you.
I'll
Keep the audience engaged.
Yeah, I'll show you the entire story tomorrow. And there was
Yeah, thank you very much. Maybe for the uninitiated in the audience, like how would you describe sample preparation in the thought process that goes into like an SDXM sample versus a XRF sample, and yeah, what's the question you have for a potential collaborator, for example?
so I'm not
Like I
sure
was
I
like
understood.
we're like you're investigating uranium, for example, and you take the samples you decide what sample thickness you have in accordance to what concentration you have in the sample, for example.
Yeah. Okay. I see the point. So all the you have a question, but you don't know how to answer it. It takes obviously it takes a lot of time and trials because as you have seen, you have your sample, it's contaminated. You think, of course, you will be seeing something, and you don't see anything. Well, I've shown you the results from Berkeley, but we've been trying samples in Berkeley for 3 years now with nothing. We see nothing because it's just bad luck. We are just not on the right size, and it's very. I think the most difficult point, and beamlines fully dedicated to imaging try to address this question, is to combine optical images, so what you can see, really, with what you put in the beam, and you need to be sure that you are on the same spot or the same localization. You need to know where you are because, and you need to, at the end, I think you need to be lucky. Because as I said, we've been trying samples for 3 years now in Berkeley with 0 results. Because each time we know there is uranium on our sample, when we go to Berkeley, they don't see anything. So it's the same, you know. But why is that? It's maybe because we are too dilute. It's not the right scale. Maybe it's not the right scale. It's like, finding a little fish in an ocean. So maybe it's not the right technique to start with. Or we are not on the right cut. Because you prepare your cuts and then you never know. And then you do TEM on your cuts. But you're never sure that it will be exactly the same as the one you bring to the beamline. So you have to do a lot of trials for the larger samples, for the geochemistry samples. It took us more than a year. Yeah, for those samples, it took us more than a year, and we had a lot of beam time. I mean, we have this advantage; we can go whenever we want, almost. To find the right preparation of samples, because we need to do a slice of the sample, but then when you start slicing it, you destroy everything. So you need to be sure that what you see is real and not an artifact of the sample preparation. So we slide it in a liquid nitrogen pool, and then applied a resin, and then I mean it was very complicated, but
Yeah, so my question was actually along those lines. So, like, say I want to look at metal ions by X-ray fluorescence in a lens with cataract. So we did one attempt, as you know, but probably was not the right scale, not the right concentration. So then you move to a different scale, you move to different, try different.
you
it's
try
kind of like
different,
trial and error,
exactly,
right?
you try
So-
different. So now for the example, I told you about that didn't really work at Berkeley. Now we are going to TEM, and I mean, we did some it's always good to do some TEM before because that's easier. So we are going back to the TEM and trying to do different seams as well. You can do peaks as well. Yeah, but there is no miracle. You think, oh, this technique will work, but even I mean, my student was in peaks in Bordeaux last week, and we thought it would be very smooth, and on some sample it's very good. On the other ones, he told me, well, seams is better. So you really have to try a lot of things. Yeah. It's difficult because you need a lot of access. You cannot, I mean, unless you really know perfectly your samples. I mean, my experience is you go to the synchrotron to do images, it will not work. Okay, the first answer, it will not work. And then you have to go back, and then you have to, really work it out. But yeah, it's more difficult than just exams. Yeah.
Now with the words, for example, European dots.
Yes.
Is luminescent or is fluorescent?
Okay, so
for example, this X-ray is fluorescent or is
no,
luminescent?
so okay, europium is fluorescent, but not in the X-ray regime. It's fluorescent with a laser. So we have done that, but we haven't done imaging.
When we can use luminescent?
You can use a luminescent or fluorescent. You can use a fluorescence probe when your atom is excited in so it can be in the red, for instance, or whatever. When your atom is excited in this energy range, and that's pretty rare actually. Europium is fluorescent, but iron is not. It has to be excited in this energy range. So it's exactly the same process that I showed for the X-rays. I mean, that you do with a visible photon. Okay, so it's your photon is excited, and you create a hole, and then you have a relaxation and an emission of another photon. Okay, but it has to be not in the it has to be in the visible. If you want. And on top of that, for fluorescence spectroscopy, you need a time, a relaxation time that is large enough so you can see it. Because if it's too small, then you won't see it. So time, for instance, time, how do we call that? Time resolved laser fluorescence spectroscopy, you can do it only for elements that are fluorescent in the visible light or close to the visible light. It depends on your laser. But I haven't seen any mapping with that because the laser beam is too large. I think.
When you do, thanks for the presentation. When you do an imaging with a certain fixed beam light frequency, how do you select that energy? It's like something fixed for some determined metals, or
Yes, actually, you a priori you choose the energy above the edge of the elements you want to look at. Okay, so if you for the images with uranium, we were above the uranium edge because we want to see the uranium. Okay, so you go, and that's really the beauty of the synchrotron, is you combine the energy tuning with the imaging. Okay, so you go above the edge of the elements you want to look at, and then if you see something, then you're happy. Okay, and then you can go below the edge, and you should see nothing. Okay, and you can subtract both image, and that's a subtraction image. And so you can do that with different elements, and then you end up with what we call correlation images, where you can correlate the localization of iron with whatever, copper, or so you can do that as well. Nanoscopium, you can do that. You with the same image, you tune the energy below and above the elements, and you subtract those images, and you get a colocalization map.
thank you.
If it works.
Thank you very much for your talk. Is it possible to do speciation on nanoparticles?
Yes. Yes. Yeah, that's 100. This one is 100 nanometer wide. It's pretty large for a nanoparticle, but we can go down 20 on this beamline. We can go down. I think the real maximum that they have ever achieved is 10 nano by 10 nano.
Just quickly, before, an error we had was if you have nanoparticles, you need to usually you don't do them, at least if you do them in transmission, you don't do them in solution in the drive. Then you need to be sure that you have nothing that precipitates or crystallizes apart from your particles, because otherwise you're literally searching the needle in the grain sack, so to speak. So there's if you want to do nanoparticles, don't have anything in your buffer that might precipitate, or otherwise just a mess of dots, and you try to find the right dot if you don't. So one more story.
And thank you for the question, because we do have a community at Synchrotron doing nanotechnology.
Yeah, but the main point with nanoparticles is. But also the question is, why would you use a synchrotron to do an image of those nanoparticles? Probably you can have a very good image with electronic microscopy.
I'm thinking, like, if you're trying to figure out how your nanoparticle interacts with a tissue, for example.
That's
like,
different point. Yeah.
Like we have
But
virus-like particles.
But you could do XAS on a pellet of nanoparticle. You don't need to separate them. That's what I mean. You see.
Maybe you're unaware that your sample has is heterogeneous in size. Each size can contribute different.
That's true. Then you need to select exactly what. Yeah, that's true.
And then,
for example, one of my colleagues is working on dope nanoparticles, doping outside thalarium with europium, and they want to know how is the europium in the whole nanoparticle.
Yeah.
And they're trying to use SAS, but they are starting. So
Yeah, that's exactly right. That's a way. That's an interesting way of looking at it. It would be to select, but you have to be lucky again. You have to select one nanoparticle. You have to find it, and if you're lucky, then then you can obviously see where your opium is and which speciation.
thank you.
We have colleagues using nanoparticles to detect, let's say, a virus in the tissue.
Yeah.
I can imagine if we want how
Yeah,
the
yeah.
nanoparticle gets absorbed or not, or distributed in tissue.
Yeah,
A
yeah.
good technique.
But my experience with stick sunbeam line, which is not that long ago, I haven't done that much, although we spent many years there, but is without luck it doesn't work. You go fishing, and I mean, we've made trips to Berkeley for absolutely nothing. It's quite, yeah, it's quite frustrating. If you have an electronic microscopy lab just close, you could go there. But of course, it's not that easy because you are at home and you have a microscope, but then you go to the synchrotron and you have your beam time, and then you have to wait for half a year.
So from this thing that you just said, so would you recommend doing like the cuts at home, looking at the electron microscope, and then those same cuts take to the
Yeah, but the danger, that would be ideal, and we've been trying to do that. We do the cuts at home, we look at them with TEM, and those grids we bring to the synchrotron. But the problem is we burn the sample usually with TEM. And so you're never sure that the sample you will see as a synchrotron is good. So what we did is we did a cut, TEM, and the next cut to the synchrotron. But it's not a proof. Yeah.
So I have a little question. When you are using the start of your experiment, how can you define what is the best beam line to use in your experiment? I suppose your college can suggest the line, the beam line, but if you are not very experimental research, how can you decide this is the best way?
Okay, I have a good answer and a bad. And the best beam line is where your best friend is. Yeah, because he will help you, So sometimes the best beam line is not the best beam line for your experiment, but that's where you can get help. Okay,
So
well, that's the way I do it.
Make friends with Christoph.
Yeah. Well, that's my view. I mean, I might not right. I don't know, but we've been doing imaging on the Mars beamline because we cooperate with them so much. But it would be better on nanoscopium, of course. Okay, but we have some other issues with safety and stuff like that. But I still go to the Mars beamline. I could go to another beamline, but I go to the Mars beamline because they are friends. And so when we start a subject with them, I like to finish it with them and not to go somewhere else. And so, of course, if I really need to, I would do it. But and so in Berkeley, the stick sunbeam line is the same. I mean, I'm not going to Hermes, which would be much closer, support as well for the radioactive stuff and etc. So that would be my first answer. Okay, you do whatever you can, but you go where you can get help because it's difficult to do. Now, of course, you need some beam lines sometimes that you don't know because you need the characteristics of the beam lines and you don't know the people. And that's it. When you went to Hermes, I mean, I didn't know them. Although I knew very well Soleil, so I could, but you didn't know them, and that's life. And then you start but then you start a new collaboration, and it takes time. The way I see my relations with synchrotrons is I almost never did measurements on a beamline where I still don't have any contact with. Meaning that a synchrotron is not just a machine where you go and then you spend 2 days and then bye. It's too complicated. Other people might have a different view, but it's important to get support, and it's important, I think, to get a real relationship with a beamline scientist, so he can help you the best he can to understand your subject, and then you come back, and of course you understand the beamline better, and you work it out, and so on one shot, I would say, unless you do exafs, and it's easier. I mean, technologically, there is no issue. I mean, depends on which sample, but basically, it's so you can go, you get your exaf spectrum, and then you go away. But for techniques that are more difficult to implement, to tune, with really challenges, I think it's better to build on a real relationship with the beamline. I think. I mean, that's my view. That's my view. Yeah.
Any more questions? No. Really good copy. Well, if not, let's thank Christoph for a really nice
Thank you.
talk. This is not in the program, but we thought we would do a group photo. So please, before the coffee break, we'll exit on this side and then around the corner where there's some stairs. And then we can easily just stand there and take a picture. And we'll come back for those of you on YouTube. We'll come back whenever the program says that we're back after the coffee break.
Nada más aquí le muteamos su
Hay que
audio.
mutear, hay que mutear,
Sí,
sí.
todo. Lo prendemos. ¿Para qué va?
¿Quieres un café?
Y le pongo.
Y ahorita le pagamos para que no.
Muy bien. So our next speaker is Mario Delgado Jaime, and I will tell you a little bit about his story. So he tells me that he's a chemist from Spirit and training and so on, but I don't know why. I just learned he studied chemical engineering at the University of Guadalajara. So that was a big surprise. I just learned. But then he went and do his PhD in chemistry at the University of British Columbia with Pierre Kennepo, right? So that's when he fell in love with metals, I guess, and protein. So and then he moved on to do a postdoc with Serena De Beer at the University of Cornell, and then moved with her to the Max Planck Institute for Chemical Energy Conversion. Then after that, he did a postdoc in the University of Utrecht in Holland. And then luckily for Mexico, I would say, since 5 years ago, he decided to come back, and he's now a professor at the University of Guadalajara, to say. And he will tell us today about the electronic structure of transition metal complexes. And now everybody should know what this means. From LHXAS and RIX 1s2p using multiplet simulations. So thank you, Mario, for coming, and also for bringing all your crew over to have you at Silvestre.
Thank you so much for the. Thanks a lot for the introduction. That's very nice, and also happy to be here in this first workshop. I divided this session in two. So the first one is just to get an idea or my perspective of how I see we can address the electronic structure problem. And in the second part, then I'll introduce what is an echo? Okay. There was some double volumes or something. And then in the second part, I introduce a program that we use to. So is my understanding is now being used more in the educational part, but it's also useful in the research part. So first, I would like to introduce some a nomenclature for understanding electronic structure. So I'll describe here one view of how we can look at electronic structure of metal ions, and this is through the term symbols. So looking at this example, fortunately, I don't have a pointer here, but the one that is here on the top, with 2 electrons in the 3D shell. So there will be, yes, yes, I have to, yeah. So there is 2 electrons in the 3D shell, and they will be able to move in these 3D orbitals because they have the same energy. And that we associate that with orbital angular momentum, which means we could be hopping between those orbitals. And since there is 2 of them, the angular momentum of one and the other will be added. So we say they are going to be coupled together. But in addition to that, we have also the spin angular momentum. So both of these electrons could be up or could be down, or one could be up and the other one could be down. And so that translates into spin angular momentum. So these two have they are described by these quantum numbers, which will be the S capital and the L capital. Oh, by the way, if there is any question along while I'm explaining things, please let me know, and we can stop and then we can discuss further. So once we have these quantum numbers, they describe very well the situation for 2 electrons or for more electrons, let's say for the poly electron, in terms of angular momentum. And they describe well the interaction of the 2 electrons. But there are more descriptions to that. So I should probably say that we are gonna be describing things in this language. So we put the multiplicity of the total angular momentum, the total spin angular momentum S. So we will put the multiplicity of that. That means the values of ms that we have available given an S value. And then we also put the letter corresponding to the orbital angular momentum. So that's just let's say a convention, right? Like let's say agreeing on that. And then, so this is just an example with 2 electrons. This is another representation of the same problem. So this is the uncoupled representation. So that means that we look at the electrons as if they were independent. So here, for example, so we put this configuration and then we have 4 possibilities. And they could be arranged in, so this description is telling us the value of ML and the sign is the value of MS. And so, so in this case, we have both the up, so they will be positive, and they are both in orbital that are ML equals to 0. So in total, we have 4 of them, 4 states for this configuration. And the coupling of these two, the individual angular momentum will give rise to this total angular momentum, which is only possible L equals to 0 because there is really no, like if there is one orbital, there is just no degeneracy there. So it's just 1, or there is a degeneracy of 1. But for the spin is where we can have 2 combinations. So one if they are subtracted, like opposite from each other, and one if they are added together. So we'll have S equals to 1. So we have 2 possibilities, the singlet, which will be from this state, S equals to 0, from this total spin angular momentum. And from S equals to 1, we'll have a triplet. And both combinations with L equals to 0 will be possible. What does that mean? That we will have 3 of the 4 states with 1 energy and the other one with a separate energy. So that's the way it's convenient. That's why it's convenient to group them in terms of total angular momentum or spin, orbital angular momentum or spin angular momentum. And this is what is represented here. If we didn't have electron-electron repulsion, all the 4 states will be the same energy given by the sum of kinetic energy and potential energy with respect to the nuclei, the position of the nuclei, and they will be degenerate. But since there is repulsion, then we have a separation of these 2 states. So this description explains very well electron-electron repulsion. However, in chemistry is more useful other representations. So let's call this a base. So this is a base, basis, sorry. So in this basis is good to describe angular momentum, but in chemistry it's better if we use a different base called basis, sorry. Well, this is just another example. Actually, maybe we can go through this quick. I'll present first the other one and then I'll come back to this one. So here we introduce the concept of crystal field parameter, which is more suitable for describing chemistry or expressing the electronic structure in these terms. So this parameter 10dq will be the result of applying electrostatic potential in the, which is the surrounding charges in an octahedral, for example, symmetry, or tetrahedral symmetry. And so the consequence of this will be that we have two groups of orbitals, and we have this description or these representations, these labels, XY, XZ, and YZ, all together transforming as a irreducible representation P2G. And we are not necessarily, except for this one, we are forced. So every single orbital, 3D orbital in the metal ion is actually, it goes an angular part and a radial part always. And so in the case of this orbital, it's a linear combination of spherical harmonic. Spherical harmonics are valid in spherical symmetry, but they are forced to mix in other symmetries, such as in this case, is not possible to, so it's no longer valid to have only one harmonic. It is a consequence of this octahedral crystal field that we have to have them mixed. And when they are mixed, they also have the other linear combination for this other orbital. So this description of the orbitals that we always learn in the university is because it is just it is a result of the octahedral or the tetrahedral, which are the two most common symmetries that are taught in organic chemistry course. But they are, I mean, they are the result of this interaction. So the other two are basically transforming together as the irreducible representation EG. And so the most stable ones are this set, and the least stable will be that, and it's exactly the opposite in the tetrahedral case. This will lead to several situations of spin state. In this case, it's only two possible for the octahedral. Depending on the precisely depending on the interaction of the electron-electron and the crystal field, which will be the opening of these two, there will be more likely to have states that are populating first this or populating both, depending if it is expensive to put them together in the same orbital, because that will cost us more repulsion between the two electrons. So the 10dq is then the separation between these two sets, and it's also the reverse. So it's the 10dq of a tetrahedral complex is minus 4/9 of the 10dq of the octahedral complex. And so moving on, actually for tetrahedral is a little bit more complex than what I just said, because there is a possibility of having mixing with the, and this is, we will see it very often in tetrahedral complexes. So this description is, let's say, a bit incomplete. So this set that transforms as a T2 irreducible representation in the tetrahedral symmetry could interact with this set in the metal ion. And so when they interact, we will get one set that is stabilized and another one that gets destabilized. And the orbitals now look, for example, if this is XY mixing with a P set, will look like something like this, which maximizes the overlap in a tetrahedral complex. So it's more oriented towards the position of the ligands will be. So that helps a lot to increase the overlap. Yeah. Question.
Okay, I'm dusting off the old lessons about hybrids. So when we look at the blue one or the magenta, is that, how would that look in a tetrahedral complex? So like,
yes,
you know what I mean? Like, where is how is that? How?
It's hard to see here. I mean,
Yeah, where are the ligands?
So here is one, here is another one, and then here is another one, and then behind is another one. So it's like this, right?
Uh-huh. And so it's more like a, okay.
So it's like the tennis ball.
Yeah. Thank you.
And then this one looks more like 4P, and then this one looks more like 3D orbital, but they are both mixed, so they have character of both. The thing is that I draw it too faint. That was, but here is a line, and then it's just that yeah, it's not. I mean, if we look at the computer, it looks much better, but yeah. So what will be the consequence of this? That there will be a less of even less splitting between the two groups of orbital. So it will be even so tetrahedral is always high spin, but there is a possibility of actually switching let's say the reverse order, so to get the order that we normally will get in octahedral if the interaction is too high. This has consequences, for example, of how the spectral will look like. This is simulations with in the KH, and the blue line is with no hybridization for a tetrahedral complex, whereas, for example, the one so it starts to look little by little. So this one gets closer and closer to this one, and if it is enough mixing, this one could go lower and reverse the order of how you will assign the electrons. For example, if you have 6 electrons.
So I'm impressed. So 4P mixing can actually make the T2 go below the E?
Not in a regular tetrahedral, I will say, like if it is special cases where you have like a very strong sigma donor, then you can do
Like,
things like that
like can you
to
give an
maximize
example?
the overlap.
Can you give an example of that? Like a complex?
I guess I don't have data to show on the KH. But I don't know if the sulfur ions that are tetrahedral are like this. I don't remember, but it could be an example there. It doesn't matter that this one is about 12 electron volts, and I think 20 for iron 2, and around 20 electron volts for iron 3. However, the component that mixes the two of them is this parameter, and if it is too strong, then it could reverse the order, for example.
What percent of 4P mixing?
I don't have the number. That will depend on this value, right? Let's see here.
On that value or the Q?
Yeah, with respect to DQ. So in this case, let's say we have a 10 DQ of 1. When we have around, let's see, so it's the red curve. So probably this one is already inverted. So it looks totally different. And it's similar to what you will get in an octahedral, which is the other state already. So it's around 3 electron volts, so that has to be huge. I don't really know if there is an example of this amount of mixing, but I will imagine it's in a very covalent that wants to maximize this overlap between the hybrids and the orbitals of the ligand.
So in your figure there's the yellow curve.
Right.
That what you showed where it has burn.
Right.
And is that only the particle part or does it
Includes both, yeah.
because when it burns, must be part of the heat counter in between the orbit.
Right.
So then how come that you still see actually the difference between particles? That is exclusive particle. And so the dipole should, I would have expected would completely take over. So that the difference into the E space, but
It has to do with the delta value. It's too high. So
yeah,
it-
but the in order for them to switch, you must have a huge overlap between the four.
Yeah,
peaks,
yeah.
Which means you have a huge amount of character in the two.
Mhm.
Peaks, basically everything.
Mhm.
So then I wonder why you can still see the transitions into E's. They are exclusive.
What do you mean? Like this set here? The transition to the E orbital? Or
Yeah, you see two peaks. the
Yeah.
yellow,
yeah.
And I would have thought one is P, the other one is sorry, E, the other one is T.
Well, I guess there is spots available in both
Yeah, but the
And-
T2 has almost all
dominating.
a lot of dipole. And it's 100 times stronger, so why is it still C?
I will say that that in both cases we have, well, in one is, as you said, is dominating the intensity, like the dipolar transition. But you still, it could be that it's just the broadening that I use for this particular case as well. I mean, I don't, and it looks like both are, both states, it has to be this one. So below, so this is, the other thing is that this is just an approximation, right? Like to assign a peak to a particular set of orbitals. So it might not be that valid here because we have several states here. It looks like there is at least another state here that is also mixing with the 4P. But it's not, it doesn't belong to completely pure E orbital, let's say. Because that's anyways an assumption, no? Like often we do this assumption that the peaks that we see in the KH, each peak is one type of orbital, but in reality they are both mixed. Through LS coupling or through, multiplet effects. Especially the LS coupling could be in here. Play a role, mixing with other nearby states. But okay, we can maybe take a look later to this simulation. It's an interesting simulation, but anyways, this is not a real example, but this is something that definitely will lower the space between the two orbitals and will also, basically the splitting of the two orbitals will be a lot smaller in this case. And then we have also the tetragonal symmetry, which was made use of two additional parameters, the ds and the dt. Now the ds, when they are positive, means that we are elongating along the Z axis. And if we compress, they are both negative because they are evaluating these integrals. A is the position of the ligand. A and B. B will be in the axial position and A will be in the XY plane. So either they are both positives or both negatives when we go from a perfect octahedral to a distorted octahedral, which is elongated or compressed. And if we keep this elongation, then we will reach the square planar situation. And then there is the limit in the limiting in which we no longer have these set ligands, then this parameter is equal to 10 dq times 2/35, just based on the definitions of this integral. So finally, for the trigonal symmetries. I mean, we are talking about here idealized symmetries, but I will put later an example in which we have a very distorted site, which is often the case in proteins as well. And then here we have the description of three parameters, very similar to the tetragonal case, but they are defined in terms of an angle, which will be the difference angle between this bond angle and 90 degrees. So for example, if it is 90, then it's exactly 0. Alpha is exactly 0. And these parameters are depending on this alpha parameter. So for example, when it's perfect octahedron, so in this case, this distortion is happening through the faces of an octahedron. So it could be also compressed or could be elongated. If it is compressed, then we will have a positive value of alpha. If it is elongated, it will be negative value of alpha. And so depending on the value of alpha, we'll have different relationships. For example, here it's possible to have a negative d tau and a positive d sigma, for example, as opposed to the case of tetragonal symmetry. So this is just the description of the different crystal field parameters that we often hear in organic chemistry. Sure.
In case for alpha equals 0,
Right.
that was very cubic to me.
This one?
Yeah.
Well, it's plotted inside a cube, certainly, but it's a perfect octahedral in the, so.
It is a perfect octahedral, right?
Right,
And so with me, it's 48.
right,
That's perfect.
right.
Which means we only have the cube. When I look in the figure, there was a pretty factor.
Right.
The degree 0 is
Certain
that's
value.
alpha.
So,
I'm a little bit difficulties to read. The x-axis is alpha.
right, this one, 0.
And so there we should. So then the q is a value.
Mm-hmm.
Maybe I don't understand what the prefactor is because.
Oh, it's a
sigma
prefactor.
should be then 0.
So it's a prefactor to the radial integral, to a radial integral similar to this, like this.
Yeah.
But the distance, so we only use one distance in this case. So let's say it's a radial like this. And the other two values of these two will be 0. So here is 0. The d sigma and d tau, the value of these two will be 0 exactly at that point.
Oh, okay. Okay. Then
Mm. Yeah, it's this one, the blue one is the dq, which is related to the 10 dq. And also, this plot is interesting because it, oftentimes in D3D symmetry, for example, in a distorted site of D3D symmetry, then we'll have the groups of the orbitals like this, and the energy ordering will depend a lot on what kind of distortion we have with respect to this angle. And so, for example, when we have a compression here means like going from 0 to 30 degrees, we are making a compression so we are making it more flat towards putting like two faces of the octahedron. So we are making the set square more stable, and we are making the other two. By the way, they have the same irreducible representation. They mix a lot. And that's why we see this crossing. So we start with this set here going down, and then here there is this crossing. So now these orbitals are the reverse for the EG, but in all cases they are mixed. There is mixtures of the two. So this is useful to get these kind of diagrams, like this parametrization. to get these kind of descriptions of metal ions. And so the other parameter that I'm going to be talking about, so I have described a little bit of term symbol. Probably I will go back later to the other one if we have time. Yeah, I need it for the second part. And but now we have described also the crystal field. And so there is another point of view, right? Like from the crystal field and from the electron repulsion parameters. And there is this other reduction that we do. So we have this set of parameters is called the Slater integrals, which is that is a measure of how much interaction we have between two, let's say 3D electrons. We can be calculated. But when we are in a complex, transition metal complex, then we need to scale it. So that's because of covalency. So we have parameters for electron repulsion. Then we also have a parameter for scaling these interactions and the crystal field. And with those, we can make a lot of sense to the description of the electronic structure of transition metal complexes. And I don't know if you have seen, so there is like a competition of what is more convenient to put the electrons all on pair, which will lead to a high spin state. And here, just briefly, this is now a term symbol, but it's in the new symmetry. So it's not longer in the spherical symmetry, but in the octahedral symmetry in this case. So which means that these electrons no longer have the liberty, let's say, well, here is locked anyways, but the one that is down could be moved freely between the orbitals here. So that's angular momentum, remaining angular momentum. Whereas the one in the top, they don't have angular momentum anymore. But these can still interact with the other part and all together have this remaining angular momentum, which is effectively the size of one, is similar to the p orbital. And in this case, we don't have any angular momentum of the spin or any angular momentum of the electrons, of the orbital, sorry. So this one is just one state here. And then here we will have 15 states that will have the same angular momentum of the spin and same angular momentum of orbital having the same energy. So depending of what is the size of 10dq, for example, here, then you will have either one state or the other as the most stable possibility. And that's part of what we trying to achieve. A big motivation for me is these electronic structures reactivity relationships that we often see in bioinorganic chemistry. So here an example of the O2 activation. So the O2 is very difficult to control the activation of oxygen. So first of all, in the triplet should be, so it's more reactive in the singlet, for example. So that's a certain level of activation. But then also, so this is without changing the oxidation state of the oxygen. But then going from oxygen to superoxide, so that involves one electron and then another electron to go to peroxide, or two electrons to go to this very dangerous radical, for example, or three electrons to water. So this is something that we often see in metallic centers of bioinorganic systems that deal in different way or activate in different degrees the oxygen. And part of it has to be with the outer sphere mechanism. I mean, how fast electrons can be provided or how fast the protons can be provided to either going this route, or another route for depending on the level of activation. But oftentimes, and this is not so much explored, is the possibility of having mixture of states. Because if we see the transition metal is it only, so for example, iron 3 has 252. If we look at the 3D shell, 252 states. They are grouped in terms of angular momentum, of orbital or spin, as we have seen. But the mixture of these states can also tune the reactivity of the metallic side and maybe going into a different pathway altogether. So, for example, these two sides, which is the IM group of myoglobin or hemoglobin, or the one in cytochrome P450, they have different action ligands, and then they have different degrees of mixtures of states. So these two states are actually the iron 2 bound to oxygen in a single state, or the alternative is superoxide with iron 3. So, but there is a coexistence in between the two, and how much you have of one and how much you have of the other one, then defines how the site in these particular pathways goes forward. And so the where the electrons are in these descriptions of electronic structure is very important for reactivity, for making sense of reactivity. So here I'm representing what I just described. So without the oxygen, then we have a high spin, let's say it's a tetragonally distorted in a C4B symmetry. And so it will be kind of like high spin, so it costs less energy to have this description of the electrons, this arrangement. And then when it interacts with the oxygen, so this, particularly this orbital will be the one that form a stronger bond with the oxygen, which originally have these two orbitals degenerate. And then either we'll have this description that I just said, is a superoxide coupled antiferromagnetically with a spin 1/2 of the iron 3, or you have iron 2 low spin and the oxygen in a singulated state. So that could be the other description. But maybe it's a mixture of the two, which is in this case the case. But in many different cases, we will see that these distortions that the metallic side has is it is actually changing the function of the metallic side. So it is an optimization of nature. I wouldn't say optimization. So it's an arrangement of the ligands by nature, so that it improves the functionality of one direction or another. In this case, the activation of oxygen. Then we have also the case of cytochrome C oxidase, for example, in which it's very important to have the conversion of oxygen all the way to water. And that involves also the binding of oxygen to this IM group and the transfer of very fast electrons and protons to live with water and to avoid, for example, the formation of this gas. So there is a variety of different spin states that we can achieve depending on the oxidation state that we have in the metallic center. Then we have different spin states in all of considering tetragonal symmetry, approximating to the tetragonal distorted symmetry, we could have these situations for the spin state. So coming back to the molecular orbital diagram of oxygen, for example, we will have two on per electron, so it's in a state of a triplet. And then the peroxide will have the population of 2 electrons in these pi star orbitals, which will destroy the pi bonding, for example. So starting to populate these orbitals here will actually make let's say, will activate the oxygen in a different way, all the way to oxide, for example. And that's what will happen in tyrosinase. So in tyrosinase or hemocyanin, so this is an image for hemocyanin. So what we have is when the two copper sides bind to oxygen, then what will happen is that the oxygen will go to peroxide. So that means an oxidation for the two copper sides. But in addition, we will have the population of these. So this is actually the molecular orbitals that I just show here. These last three, let's say. And so here, the population of these will start to destroy this bonding between oxygen and oxygen. And so we'll make it more easily to be transferred one oxygen to the substrate, for example, because of the destruction of that bond. And so what happens in tyrosinase, for example, there is room for, so there is room for a phenyl group to bind to copper. So it increased the donation to the copper, and it increased the back donation to this orbital in the oxygen to make the destruction even more easy once the substrate is there. So I guess what I'm trying to show here is just examples in which it matters a lot where the electrons are. For example, in this description is again, so it's a more complete picture. So it's a molecular orbital diagram of an octahedral complex, which is showing that the orbitals, the EG set, for example, will be actually anti-bonding, sigma bonding. So if there is electrons here, it will activate the metallic complex will behave differently than if all the electrons are here in the T2G, because this one is involved, in this case, in the pi bonding with the, so this is actually the molecular orbital diagram that will be compatible with ferricyanide, for example. And in this case, we will have these orbitals, the T2G set, which is a pi back bonding to the cyanide ligands. So it's very different to have the electrons here than having the electrons here as well. And another point that I want to make, that is very often actually that we could see this mixture of states, even in, for example, let's go to this case. So here is, I'm showing how the different states that, as we described, for example, in this extreme, the different states could be described more in terms of term symbols, no? This total angular momentum of orbital or total angular momentum of spin. And as we go along with the increase of 10dq, then we start to split the energies of this state in different way. And depending on the value of 10dq, the ground state, which is always this, is always a triplet T1. but the triplet T1 has two situations. It could be used the way these two electrons are moving in this set, which makes this situation transforms the T1 irreducible representation for the angular momentum of orbital. But the other possibility is this, and these two possibilities, for example, they transform in the same way, or let's say there is arrangements here that are compatible with this total angular momentum. And that means that in a state that in a metallic complex, transition metal complex that has a very low 10dq, these two scenarios will be significantly mixed. And what it means is, so in this case, is 90% of this situation and 10% of this situation. So it's the tuning of this mixing of where the electrons are is what is making behave in a different way the transition metal complex. But even in this perfectly octahedral, we observe this kind of situation. And then if we go to the other extreme, or let's say a 10dq that is high, then this mixing almost disappears. So it's almost like a pure of this state in this other representation. So this description of the electronic structure I like because it's, as I said, more informative to chemistry problems, right? Like it's more familiar of understanding a little bit more of the reactivity that we observe. So later I will show how to generate this kind of diagram in the program and the composition in terms of one particular basis or another basis. Angular momentum. So this description has still some remaining of orbital angular momentum, the T21, because these two electrons could be moving freely between these degenerate levels. Or for example, this electron could be moving between these two or these three. The coupling together of these two scenarios give rise to the same angular momentum that in this case.
Thank you.
So, and that's why they are mixing actually. That's the reason why they are mixing. This is by electron-electron repulsion.
And this mixing can be reversible?
So meaning that
Reversible? It's time reversal?
the mixing of states, I guess, yeah, let's, I'll show you an example in which we are actually in exactly in the opposite order, because when we have no 10dq, then we have a scenario in which they mix in a different way. So I can show you later with the program.
I'm not sure just a question that you asked is a reversibility. So that is, there's no chemistry involved when they mix, just physics.
Right now is
Okay.
physics effect, yeah.
There's no reaction so going on. It's just to have electron change.
Oh, sorry, I misunderstood the question. So that means that by electron-electron interaction, there is a mixing of these two states. That means that, for example, this electron has angular momentum between orbital angular momentum between these two that are degenerate. And the other electron too can move freely here. But then they talk to each other and then they generate a total angular momentum that is equivalent to T21. And the other case, which is these two moving, they are degenerate as well, like this level, so they can be moving in any of the orbital. They also generate an angular momentum that is equivalent to a T21 symmetry.
They share the same magnitude.
So they mix because they have the same total angular momentum of orbital. So that's why Thomas said there is a physical effect that these two states are mixing. But this situation of mixing, we also see by, for example, spin orbit coupling promotes a lot of mixing as well. And also bonding promotes a lot of mixing. And distortions of the crystal field also promotes mixing, as we will see moving forward. I don't know if there is more questions so far. So how much time left? Okay, good. And so all of these that I have described so far, turns out that the LH-XAS is very sensitive to see the ground state electronic structure, or saying in a different way, depending on the electronic structure is the states that we are able to reach in a final state of LH-XAS spectroscopy. So just a quick review. It's always nice to have a refresh of what is this spectroscopy. So the LH-XAS involves the transition of the electrons that are in the 2p shell of a transition metal complex, promoting into the valence transition metal complex that will be the 3D. That will make it 2p to 3D will be a dipolar transition. And they are mainly separated by spin orbit coupling. So that means that here we have another physical effect, which is interacting the spin part and the orbital part of each electron. And here we should generate a hole when we move one electron from the 2p shell to the 3D. There is a hole that is being created that will be sensitive to this very strong spin orbit coupling. And since it's from the 2p shell, it's going to be significantly strong. So it will be the strongest interaction that is observed in the LH. So it is sensitive also to the crystal field, as we will see. And because of the other secondary interactions, which will be the electron-electron and the crystal field, we will see different shapes. And there is also a contrast of what we will see in the ground state and the final states. And that's why we see very different, a lot of states being probed in this spectroscopy. So I just want to show you. So here is just a description of how we, for example, calculate a simulation to try to make sense of what is measured in the synchrotron. And so this is the total Hamiltonian, which will involve the kinetic and potential energy part, and then the electron-electron interaction, the crystal field, the spin orbit coupling. This is in the 3D shell, for example. And then in the final state, after the transition happens, this will be a larger Hamiltonian. So these matrices are actually the interaction of every microstate with every microstate in this final state. For example, for iron 3, that will be 1512 states, microstates. So it will be big matrices of that needs to be solved. And once we solve for the initial and the final state, we will have a ground state here in this configuration, and we will have many states in the final configuration. And so there is this, let's say, electric dipole transition matrix, which relates every single state with every single state, every microstate with every microstate. And so once we solve the Hamiltonians of each, the ground state and the final state, they are mixed with all these microstates. And so that's why we, from here to here, then we have like a projection, which is actually the intensity that we see in the spectrum. And this is actually, let's say this is a calculated spectrum, and then it's broadened to actually have the same shape and bandwidth of the experiment. But so this is basically what we do for creating a simulation. And so from these simulations, so we could see, as I said, LH is very sensitive to the ground state for this reason, because depending on the different interactions, then you have a different ground state and then a different projection here. So I'm showing here two cases. So we have a situation where we have a transition metal complex, which is high spin, 10 to the cube of 1, and then 10 to the cube of 3. And the spectra is very different one from the other. So I'm showing two different broadening. So it's the same spectra, but different broadening. That if we don't have too much resolution, it could be a little bit more complicated to actually extract the electronic structure. So, and here is another example, but here we have two parameters, 10 to the cube and dA. So 10 to the cube is the opening of the octahedral crystal field, and the ds is mostly for tetragonal distortions. So it's either for, as we saw before, elongation or compression of the metallic side. And so depending on the combination of these two, we will have a different ground state. So for example, this one in orange is this ground state, which is a high spin, and also the one that is blue is also a high spin. So these two are high spin. And then if the 10 dq increases, we have a different ground state. And we in tetragonally distorted complexes, we will have also the possibility of intermediate. So this is kind of like a phase diagram of combination of 10 dq and ds, which can lead to a intermediate state. And from these ground states, different ground states, we obtain a different, completely different LH XAS. So it's extremely sensitive to crystal field effects and multiple, especially crystal field, because of the shape that it arises from the different cases. And then, as I mentioned, spin-orbit coupling, it's another example where we can have this mixing. So here we have a situation in which we have this, this is for cobalt 2, 3d7. And for the state quartet T21, which is lower values of 10 dq, is the ground state, is actually a mixture of these two configurations. And again, looking at the two states from the point of view of chemistry, we have two electrons in Eg orbital, antibonding, sigma antibonding, and here we have three. So this mixture is because of electron-electron repulsion. But in addition to that, these two are related to these other states by spin-orbit coupling. And it turns out that very close to the spin crossover, which is to a certain spin pairing energy, we will have maximize the mixture of these two states. So in this case, for example, the ground state is the mixing of these three configurations and these two states. And so that will lead to a very different shape. So here, for example, is a low spin cobalt, and here is high spin cobalt, but here is kind of like a mixture of the two. And we can capture by LHXAS spectroscopy this situation. There was a question, no, right? And then I want to show this example, which is other crystal fields, related to other crystal fields. So this is a mineral, which is a manganese 3, but in 2 different sites. So this is the crystal structure. Well, not the crystal structure really, is more where the locations of these, the manganese sites are in the crystal, because I'm not showing the oxygen. So the ones in green are very ordered, traditional octahedral site, but the one in red is extremely distorted geometry. So it's not really, we cannot say it's really tetragonally distorted, or we cannot say it's trigonally distorted. Is something similar that we will find in proteins very often. So this is in this particular material. And so what if we do a simulation of this side and in this side? And so this is the experimental here. And this is the sum of the two. But look how different in one side than the other is. If we analyze, so what is the strategy here? So first is to interpret with simulations let's say the LH, or it could be, as I will show you in a couple of minutes, the RIX 1s2p. So, but once we have a successful simulation, now we can analyze the ground state. And in the analysis of the ground state, we found the following. So this will be the crystal field diagram for the octahedral site, which is a quintet E state. But in the highly distorted side, we have this crystal field diagram, which is probably nothing like there is this is closely degenerate, these two levels. So let's say this one is similar to a compression in an octahedral site. And then, but then this leads to a very well-defined ground state, which is quintet T2, very well separated from the next state. But what is interesting is that this quintet T2 is impossible to stabilize in a perfect octahedral. So this is more like the ground state of a tetrahedral, actually. So this one we'll find in a tetrahedral, and this one we will find in an octahedral. So here we have a site that has to be octahedral, but is distorted to have the ground state of a tetrahedral. And that is also reflected in the mixing of the different spherical harmonics that build every single orbital. Turns out that the one with the highest energy that interacts the most with the charges in this side has almost exclusively 50% of this and this, which actually is a linear combination that is required for the XZ orbital, for example. So it's as if we look from the perspective of a tetrahedral, is as all of a sudden one of these orbital, which is the XZ orbital, will go very high in energy because of this distortion. And that will change completely the electronic structure of the site. Because so it's kind of like, that's why I said that oftentimes in proteins, these distortions lead in purpose to a different ground state so that it actually has the functionality that is needed by the biological system. And so just one quick mention of in proteins, we cannot get LH XAS because of the vacuum conditions. But we can measure what is called the RIX, the resonant inelastic X-ray scattering, which will be, let's say, more or less equivalent. So it's in a strategy to get an LH like spectrum when we go in two processes, when we mix two processes. The first one is starting with your ground state, whichever ground state is, has the protein, let's say. And then we excite to, so we move one electron from the 1s2 to the 3D shell. So now we have 1s1, 2p6, 3d6. And then from this intermediate state, depending on which level or which state was reached by the X-ray, so that will be the incident energy of the X-ray. From there, there will be a different projection into the final stage, which will be a decay from the 2p shell into the 1s. And the final state and the ground state has the same kind of relationship to the LH. And so we can probe similar spectrum, so we will obtain similar spectrum than the LH, although we can have a different kind of projection depending on which state we are exciting to. So that will be the experiment to, for example, use in proteins, no? Because the ultra high vacuum will not make this possible.
We can understand this final state as a kind of density of states when we for solid state physics.
I will say that, for example, here, as Thomas was saying earlier, so this is an approximation though. So we assign oftentimes, so this is what we will see in the KH. So this is the excitation energy. And oftentimes, some of these features are assigned to a different group of orbitals. So it could be the T2 or the EG, for example, in this nearly octahedral. And so from here, what you will see is like is close to what will be, let's say, in a regular LH. If you could decompose your LH, it will be like whatever transitions are happening related to that orbital. So it's similar to the density of states, right? Like it will show the projection that is related to one particular kind of orbital. So it's either EG or T2G projection, almost like a decomposition of your LH, depending on the. But this is also an approximation. In certain cases, it's a valid approximation in which you assign one particular feature in the KH to a particular orbital, but in some cases, this might break down. And it's just a mixture of the two groups of orbitals, and then you will have contributions in the two excitations. Let's say you take one cut for the measurement of the emission, and here, then you will get the red one, and then you get the green one if you actually measure at this excitation energy. So you get, let's say, different projections depending on the nature of the orbital.
Thank you. Moreover, you say that there are so many ground states because you have several electronic configuration.
This one.
So is it possible to say that there are metastable electronic configuration or not?
Sometimes you have excited states that are very close to the ground state. And in the spectrum, the experimental spectrum, they will be populated by Boltzmann distribution, and they can also contribute. But this is also very interesting because when we see this multi-state picture, like the ground state and all the excited states, then this is like different avenues of reactivity coming together. Or that's the way I would like to think about those. Like you have so many possibilities nearby for going stabilizing one ground state or another, so they are very close together, and so you have more possibilities right there, no? And when they are very close, you could assume that they are actually mixed, which is happens a lot of times for different systems. Kind of like in purpose, like the one I was describing of having different couplings of the unpaired electrons in the in groups, for example. That's one example in which we can have that situation. So I think I think we still have time, no? Like, yeah. So now I will like to go to the second part, which is more related to the analysis of the ground state. Let's say we have one kind of spectroscopy, it could be LH, KH, RIGS, and you have found a successful simulation. We can now analyze the ground state, for example. So we have this tool, CTM for DOC, which I will show. So by the way, CTM for DOC is a program that can be downloaded from the internet. So this is mostly for Windows users. So it's a standalone. I probably will increase the size. Actually, I need to share in a different way because now I have to share it like this, because I will be switching back and forth between this and the MATLAB. So this is a software that is in this version is a Windows version. And then we have also the MATLAB version, which is an effort to make it all the calculations faster, because this is code used from a long time ago, was starting to develop in the 1970s for multiplex simulations. And now we are using our own code, making some of the calculations that you will see much faster. So if you have MATLAB, later we plan to compile as standalones as well, but at the moment, still kind of like in the development process. This one is totally usable, so you can use it if you go to this. So it will, maybe I will send this later so that it's just one click for the people that register. I can send you the link. You can download and do these calculations. Well, let's look at this example just to see what is what this program. I'm not gonna cover all the things that this program can do, but if I'm able to show a few examples, that will be great, because we have a little bit of limitation of time. So let's go to a 3D2 configuration just for this example, and then we switch to other example. So in the 3D2 configurations, then we have the coupling of the two angular momentum of these two electrons will result in a total angular orbital angular momentum of 0, 1, 2, 3, and 4, which is S, P, D, F, and G. And in combination with this total spin angular momentum of 0 and 1, which will lead to singlet and triplet. So some of those will be not allowed by the Pauli exclusion principle. So the ones that are allowed is the one with that has a sum that is even, uses because of the properties of the antisymmetric wave function for the fermions. So that's so there is a method to calculate all the states for these term symbols, but in total we will have 45 different microstates grouped in 5 different energies in the end. And so you will see that we can actually calculate all these 45. Let's do this. So first once you download what I'm gonna send in the link, you just go to the folder and type this. And then a window like this will show. And so here we can, I'll put vanadium 3, which has 3D2 configuration. So these 2 parameters here is electron repulsion. So I'll leave it at 100%, their value. So this is the atomic value for vanadium 3. I will have a 0 spin orbit coupling, and I will have a 0 10 DQ. And with that, we can calculate so basically we have only electron repulsion at this point. It's just a metal ion by itself. And then we do an example of vanadium 3, 2. And then we can see how the states are grouped. So it's 5 energies, but they have degeneracy. For example, the first energy is in total 21 of them. And it's 21. If we go to atomic term symbols, we will see that it's 21 because it's a triplet F. So is the degeneracy will be 3 times 7. So it will be a total of 21 states like this. And so this state, for example, is very well represented by a basis that uses these term symbols. So what will be these 21 states according to this? It will be the several values that we have for MS, which will be -1, 0, and +1 when we have a triplet, and values from -3 to +3 when we have L equals to 3. So it's every single of these states are those states, but sometimes in the microstate representation, you have mixtures. So here, for example, we have this microstate. It's hard to see, but it's -2 plus and -1 plus. And that state, oh, I think this one got moved. Oh, I see. So will be this guy here, right? So this is one state that is pure. But for example, when you have the same value of MS and ML, then you will have a mixture in this description, and you will see it here. So let's move to a different state. So you will see at certain point you will have a mixture. So here, for example, they are mixing 2 of them, because this mixture is exactly correspond to another state in the other representation. So in this case is triplet F, ML -1, MS -1, for example. And this state that is represented by a single representation in this basis is a mixture of microstate in the other basis. And so here you can explore what will be the linear combination, for example, of cubic crystal field, which is what I was showing before. So it's a mixture of these two states with a T2G, 2 electrons, and 0 MEG, or 1 and 1, for example. And so this allows you to basically change how you look at the states that you are generating on the certain combination of parameters that are making your ground state. So what you will put here, I mean, for research purposes, what I'm showing here right now is more for educational purposes. But for example, for research purposes, then you will put exactly the parameters that you use for your best fit, and then you will describe the electronic structure of your ground state and also the excited states according to the preferred basis sets. There are other things that you can do as well, which is, let's see.
Maybe,
Sorry.
I have a question.
Yeah.
So, right now, exactly what you said, just for me to repeat and understand. So you would go to take your vanadium sample, the synchrotron, collect the LH, and understand the splitting by that, and then go back to the program, put in the parameters you experimentally determined, and with that understanding now of microstates, go back to the spectrum in order to better understand visual contributions.
So once you collect the spectrum, the idea is to feed the spectrum with a multiplet simulation. And then it could be, normally it's done manually, but tomorrow I will show how it is, could be an automatic process now of because sometimes it's like a needle in the haystack kind of problem. So it's so I'll show you that method tomorrow. But once you have that, then you use exactly the same parameters, and then you get a description of the ground state here. And so this is a different point of view of the 445 states, for example, in which only we will have 10dq and no electron-electron repulsion. So in this case, we have these three cases. So we have the 2 electrons being in T2G, or 1 in T2G and 1 in EG, and the other, or the 2 electrons in EG. And this will be the degeneracies of each of them. So this is something that we can also calculate this case scenario. But what we are gonna do is to try to reproduce, for example, this diagram, Tanabe-Sugano diagram, or an energy diagram, which we could employ also with techniques of UV-based spectroscopy, for example. So we are going from the atomic, completely atomic, all the way to a scenario of a very strong crystal field, in which we have this almost all the states aligning to these energies, these three energies. So to do that, and this could be quite useful to find what will be the best fit to a UV beast, for example. So we can do a Tanabe-Sugano, in which we move the 10dq. In a step, let's say, of 0.2, so that we don't have that many points. So we could see here how this, if we go to a even higher, so I'll do that in a second, but you see how this electronic state starts to open and they are very described in a different basis sets now, which is the cubic term symbol. So it will be the best description of each of these states. And as I mentioned, if we look at different basis sets, like for example, the crystal field, where the electrons are, we will see a different levels of mixtures for every value of 10dq. So for example, if we go to a 10dq of 1, now we can look at a code at 1, and we will see exactly the states at this code. And we can use all the bases that are available for representing every single of these states. So this one, for chemistry, this one is, as I said, the most useful. So this is, for example, a tetragonal representation, tetragonal crystal field representation of every state, no, for example. But let's see. Let's see a real case here. So this is a cobalt complex, cobalt 3 plus. So this is the UVBs, and this is two transitions that happen at more or less 2 electron volts and 3 electron volts. And so let's say we want to find out what is the value of 10dq in this complex. So this is a typical UVBs spectrum. How will we do that in the program? So the issue here, so we have to change to cobalt 3 now. So now we have a configuration of 3d6, and we need to move the 10dq and find out where we can see two transitions in the UVBs spectra. So in the UVBs spectra, it's important to keep the spin the same. For example, if we have a low spin state, then we can only have transitions to a singlet in UVB. So that will be the selection rules, not to change the spin state. But if we have a different state, so that will be like, in this case, like a quintet state, then we will have a different number of transitions in this case. So an important part in a octahedral complex is that we have to reduce also the electron-electron repulsion because of the Neifel-Ausetic effect. So we will have to create this diagram, the Tanabe-Sugano diagram, for several scenarios. Let's do, for example, 90% reduction. And then we do a 10dq from 0 to 4 to try to look for what is this, the 10dq of this complex. So this Tanabe-Sugano is with a reduction of 90%. I don't think it has finished calculating. Oh, it's there. So now we have a lot more states because now we have configuration of D6. So we have 210 group in different multiplets. So here we have one ground state, which is different to this one. So let's reveal what is this and what is that in order to know. So we can also have, for example, if we put here 0, then we can make always the ground state 0. and everything else with different. Something that we are doing, working right now, is to classify all these lines according to the spin state. So that is easier to identify. Way more easy to identify what could be the 10dq value depending on the separation of the lines. Because right now, so first we need to identify so for example here, 10dq of 1. So 10dq of 1, the ground state is actually quintet T2. So the quintet T2 only have the chance to go to quintet E. So that will be one transition. But if we go to the other side after the crossover, let's say 2.5 or 2.6, then we see singlet A1. And for the singlet A1, we have several other multiplets like this one. So it's hard to see in this screen. But this one is also a singlet. And the separation of energy has to be the same as the bands that we see in the UV-Vis spectra, for example. So I guess this reduction of the electron-electron repulsion has to be then, so it's a iterative process, right? So we change. So one thing that we are working right now is to actually do the other way around. So you have so you know where the energies are, and then you search for your solution in terms of the reduction of the electron-electron repulsion and the crystal field. Because of course, you can also do this kind of diagrams for different crystal field parameters. For example, for the tetragonal distortion. And then if you have the enough resolutions, I know in UNAM they have an instrument that you could see very well resolved UV transitions. I think it's from Professor Nora. She has an instrument. And so you could see more structure in the bands.
Yes, the spectrometer, this UV-Vis and near infrared. So it's bigger. We can see more transitions in there.
And so it's not as broad as this one, right? Sometimes maybe, but I think I saw some spectra one time that you could see the tetragonal distortions as well. So you could potentially use search for 10dq and DS as well. And also the reduction of the Neifel-Ausetic effect, searching the program, let's say.
Yes, in the spectrum we can see that. And we calculated 10dq with the graphical approximation, so we can calculate that. But yes.
Mm-hmm. Is that the Zoom? We might have lost the Zoom. So I'm about to finish now. I just want to mention about this spectroscopy. So this is very nice spectroscopy. So it's just 2 slides. So in this case, you make a transition from the 2P shell to the 3D. Oops. We'll share again. Yeah, so there is an electron from the 2P shell to the 3D. And again, we have, so this is like the LH actually. So all these states are available, and depending on the ground state is what the spectra that you will see, that's the LH. And then depending of the excitation energy, let's say you jump to this state, you will see a different decay. So it's a decay that is coming back, simply coming back to the original shell. And so you have the same final state that in the ground state, but it's inelastic. So actually this one I should have put it the same level of energy. That's inelastic in the sense that you will see different peaks for the different multiplets. And so in this spectroscopy, you could see all the lines of the Tanabe-Sugano diagram, which is very nice. So depending on the excitation, for example, if you excite to this particular state, that will be this spectrum. So you will see some of the multiplets. But if you excite a different energy, different cut, then you reveal different states. So for example, in F, you see almost all the states of including these two degenerate, for example. So you more nicely can find the crystal field, for example. So I will leave it at that because of we don't have too much time, but left.
First, thank Mario for this amazing presentation. Are there any questions? Anyone would like to ask? Yeah, question. Maybe because this slide is up, this is amazing. I love this. Personally, I would really like to do something with that. So what are the experimental limitations for that? It's all the questions. What's the concentration I need for that? What's the
for rigs, for this kind of rigs, is actually limited also by the fact that it's actually under vacuum. So it's not, so it's great for solids and for materials that are involved, for example, heterogeneous catalysis. It could be something interesting to explore because mainly because of the conditions that you need for the LH. That will be a very important limitation, for example, to work with proteins, right? They won't.
In the spirit of, can it work with proteins? You know, just because something is not soluble doesn't mean that it is impossible to do. Could one lyophilize their protein, for example, and load it into this? Let's do it. I got plenty of protein.
I always encounter that plenty of one is almost nothing for someone else. But yeah, it would be. I would love that.
Yeah, this is a very nice spectroscopy.
More questions, ideas, comments? Morning.
So in the Riggs diagram, we have like these two axes. One is the irradiation energy, and the other one is like the energy that is emitted through the
The thing is that you are measuring two things. So is first is the absorption, or you control the excitation energy. So you jump to different states.
Yeah.
Which is what is represented by these peaks, right? Different states here. So let's say you measure at this cut, then you from there you can jump back. So basically you are bringing one electron from here to here. So you will return to a different to all these states, but with different intensity, depending on which state you're
jumping
from.
from. So what in the colored one, it's like the same principle?
The color one.
there is like this. Uh,
The
2D
map.
The maps, yeah.
Oh, yes. Yeah, this map, actually, you could see so this is a very nice thing that you could do in CTM for DOC. So this is just finding what is your ground state. So it's something that is available. So it's a phase diagram. So it's showing what are the ground state depending on the values of DS and NDQ, which will make your complex tetragonal, so D4H, or square planar. And for example, here I'm showing a cut that you do here exactly at NDQ of 2. And this is changing the DS. So here it will be a transition metal complex that is elongated or compressed. And you will change the nature of the ground state. And that will lead to a different XAS spectrum, or actually any of these spectroscopies is very sensitive to the crystal field. But yeah, this you can generate in the program.
Another question from the audience.
Hello, thank you for your talk. It was very interesting.
Thank you.
So I was wondering, I'm very interested in cobalt oxide
Mhm.
because I also work with cobalt. And have you ever tried working with hybrid materials, or have you ever tried to study the interaction between the cobalt oxide and a polymer, for example, and it's possible to calculate also the skew for this type of material, or have you ever thought about it?
Yeah, that's a good question. So I was involved once in this kind of measurement. So there is a publication on that. Maybe I should share it with you. So we did this kind of measurement for a possible Fischer-Tropsch catalyst. So it was oxide of cobalt, and it was with some nanostructure materials in there. And it was so it was the conditions of the Fischer-Tropsch catalyst, which is oftentimes high energy. And then from there, we could see the changes, not only in the LH, but also in the RIX data, as the reaction is, for example. So you will see a change of, for example, cobalt and cobalt oxide proportions, because the cobalt starts to get reduced as the reaction progresses, for example. So
Thank you.
normally for this, so you will have to yeah, for solids is easier than for proteins, for sure. Yeah, for doing this kind of measurements. And then you can extract a lot of information like your crystal field. And there is one thing that I didn't discuss, I didn't have time, which is charge transfer effects, where you can extract covalency or include, let's say, bonding into your system. So that's also very interesting, and all this methodology is useful also for that. So
Okay, I think we can leave it at that.
Mhm.
Any more questions at the lunch we will have right now? So anyone following us live can do that as well. And we will be back at 3.
Good. Of course. No, don't. I thought you were just gonna pick a couple things. Okay. Oh, the Dreyfus or the career? You want the microphones? Where's everybody? San Tomas. Sí. Nils, do you like the acronym? The SBSSMB. SBSSMB. It could work. We can brand it. Where is everybody?
When I gave my first talk to XAS here, and then it was a good question. So could you explicar luego lo de Hades?
Oh. Yeah, I agree. We have plenty of time. I can wait if you want to. Yeah. Now we can start. And Mario. So much good science out there. I can't blame them.
Okay. Okay, welcome back. We're on. Okay, we're on. Good. Welcome back, everyone here in the room and whoever is out there on YouTube. So I'm very happy to introduce to you Kelly Chacon from Reed College. And she did her bachelor's in chemistry and biochemistry with departmental honors from Mount Hood Community College in Oregon and also Clatsop Community College.
Sí.
Right.
Y luego Portland State University.
Okay, so let me start. Then
3 times, 3 tries.
Then she did her PhD in biochemistry and molecular biology at Portland State University.
Oregon Health and Science University.
I'm sorry, I'm reading everything wrong. I'm very sorry. Or
I don't
Oregon
care.
Health and Science University.
I'm here. I can
also
correct it.
Portland. Thank you. Thank
It's
you.
fine. If I weren't here, it'd be a problem. But
And since 2015, she's an associate professor in the Department of Chemistry at the Reed College. And I met Kelly maybe at a Gordon conference, Metals in Biology, over some beer at a poster session.
yes, the
And,
best way.
and then we connected. And when I was putting together this program, I said, who do I know who does really cool science and actually is a very nice person? And actually we have some Latino connection.
Video.
So she very proudly holds the Mexico-US
Oh yeah.
Chicano
Oh yeah.
flag, right?
Los Mulos.
And and I'm very happy to have you, Kelly, here in Mexico City for her first time, I guess.
Time.
And hopefully not the last one.
No.
The first one of many.
Guarantee. No, not the last time.
And she changed her title, as you can see. Considerations for, so the old title was Considerations for Biological Exo Simulations, and now she has a cooler title. I I told you she's very cool.
Oh, yeah,
Um,
so cool.
Cool way, cool ways to use BioXAS and how to get started on a beam line. And her promise is that by the end of this talk, we're going to have a little application for beam tech.
That's right. You're going to be completely ready.
So, and last but not least, just last week, as she was getting ready for this trip, she got the news that the Dreyfus Foundation gave her this Teacher Scholars Award. They only gave 8 of them, so we're very happy you have been one of the teaching scholars with this prestigious prize.
I'm going to
And
teach
we
you.
are going to
So-
celebrate with some tequila
Yes.
when this workshop ends.
I'm going to teach you
So-
so hard. Okay.
Thank you, Kelly, for coming. And
Oh,
for being
I'm so excited to be here. I'm sure that I've talked to a few of you. My dad is from Nayarit. He is an immigrant, but he still is a Mexican citizen. And so I'm going through dual citizenship. It's very important to me to have both sides because it's part of mi cultura. And also to when I got my PhD, my family in Mexico and my family in the United States, they finished maybe 5th grade and high school, you know, each side. And so to me, it felt like I was getting further away from my family, you know, to try to describe what you do. And to me, this is just such a beautiful thing to see science in Mexico, to see that part of me expressed and understood, but also my identity. So I'm really excited to be here to do this. It's just a great honor. So yeah, this is my stuff. I'll put it up at the end if you want to contact me. This is my lab logo. It's a little bit dark here, but I will not be talking about tellurium on here because I haven't published it yet. So I'm going to wait, but if you want to talk to me about it offline, I'm totally happy to pull up some slides and talk to you about it. Also, at the end of this talk, I will turn off the recording so that I can share some actual SSRL proposals with you with permission of the people, and some of mine too, like my first one, and just kind of show you what's up. So yeah, I'm very excited about that. I said, very excited about that. Okay. So some of you have already asked me, oh, I don't even have to hurry. This is great. Okay. So this is my college. You can see it looks very old and sort of special. What it is a 4-year college. So college is different from university. A college is a place where we only train undergraduates. Okay. What's the equivalent here? Licenciados? Okay. And so what I was passionate about, some of the colleges will have maybe a master's or a couple of PhD programs, but very minimal. The idea is I'm not training current scientists, I'm training future scientists. I take pride in the idea that when someone leaves my laboratory, they could join Liliana's lab as a graduate student and be able to jump right in and get very excited about things. Yeah. And to that end, Reed College is actually a pretty elite, what they say elite, which means money. Okay. So what that means is that my laboratory is almost a carbon copy of my PhD advisor's, and he had lots of funding. So I was really able to make something that is real for the undergraduates to do real research. And of course, my pride is to be able to do research that is, you know, comparable to my field, and to apply for competitive grants once in a while. So that's very important. And to have undergraduates part of my papers. I think that's just so cool. So you can kind of see it's very rainy in Portland. It's close to Seattle, if you know where that is, but basically it's north of California. Okay, let's just put it that way. Okay. So now on to the work. So this is what I think about when I think about my particular job as a scientist. Biochemistry, metals, and spectroscopy. Together, that can be called bioinorganic chemistry, metallobiochemistry, biology of metals. There's a million names, doesn't really matter. Bottom line is we really get to the heart of a metalloprotein. And the example I'm going to talk about in the first half of my talk is Thermophilus copper A, beautiful copper 1, copper 2 metalloprotein. And we use a lot of tools. Holy crap, that was loud. You guys hear that? Okay. We use a lot of tools to be able to do that. So crystallography is one that we know a lot about. Of course, now we have cryo-EM, which is coming up as something to do on more difficult samples or things that have different subunits. I'm using it currently for a membrane protein. So something that is really It really tests your knowledge of biochemistry to get something fatty out of the fat and then put it into another fat and then figure out how to keep it happy. And then if it has a metal, how do you look at that? And then, of course, there's electronic absorption / UV visible spectroscopy, fluorescence spectroscopy, electron paramagnetic resonance if you have something that has an unpaired electron or a radical, and then, of course, extended X-ray absorption fine structure, or XSAFs, which has been my passion and bread and butter, I guess you could say as well. So this goes for actually most metals, but since I'm going to talk a lot about copper, I just wanted to point out something. This is kind of what I'm obsessed, not kind of, this is what I'm obsessed with. People say kind of, and I'm like, no, I'm going to commit. I'm obsessed with it. Free copper is never encountered in the living cell. In fact, it was calculated, which, you know, it's a funny number, that there's less than an atom of free copper in the cell at any given time. And that was some really cool, rigorous science to figure that out. But the same thing is true for pretty much any metal in the cell. You're just never going to see it drifting around. The minute that you eat your food and the metal goes through your stomach, it's being held by something at every single point. And we actually don't understand a whole lot about what's responsible or even how these, in this big, you know, you've seen probably the images of a cell and how everything's really crowded together and there's a million different things. How in the world does a chaperone recognize its target in this milieu and be able to specifically make it move in a certain direction? And of course, there are various diseases that can have huge impacts on this, especially pretty much they're generally right when a baby is born or something like that, and it's lacking something. We'll talk about that in a minute. But in general, there's a couple of interesting things. But what I ask is, what basic chemistry, just fundamental chemistry, underlies that level of control? I love the cell, but it's big and it's messy. And personally, I think many spectroscopists can agree, it's really nice to get down to the nitty gritty and try to answer these questions. And so you can see a lot of what we call blobology here, of just circles of things. But this is a bacteria. And I use bacteria because I don't want to slice rat brains, for example, like Liliana had done to earn her way. But bacteria is actually, I mean, these things, again, the fundamental chemistry of these things should underlie many other processes that could be applicable to humans. And also we can use these things to fight bad bacteria. Think about it. E. coli lives in your gut. It lives by the seat of its pants. I don't know what the corollary is in Spanish, but basically, what would you think? Like the seat of the pants. I don't know. I think it's, well, just one day is this, one day is that, right? It just depends on what's going on in the gut or wherever it is. Okay, it has to absorb everything from the environment. It really doesn't live on its own. So we can think about micronutrients, okay? So it eats all the things. What it can't do is block out tiny little metals. And so one of the ideas is that if you could figure out what basic chemistry underlies all of this, that you could figure out ways to stop it. Okay, so there's a lot of people interested in that, and I'll talk more about that in a second. But it's just to convince you that this has downstream applications, but what we're going to talk about, of course, is more of the basic science. So today, 3 different stories, and then we'll continue on one of these stories tomorrow. So the first one is a green path toward purple assembly. I know I'm giving you all these fancy titles for things to get you excited. The Copper A story, telenovela style. And then we have copper and silver weapons in the host pathogen war zone. Really dramatized, the CUS story. And then, of course, at the end here, we're going to talk about getting started at a national lab. And it turns out that I'm actually an SSRL executive review panel member. I see pretty much everybody's BioXAS proposals, assign reviewers to those, read all of the proposals, help rate them. And so within reason, I can't wait to give you some pointers on how to do this. And mostly what I want to encourage you to do is just try. Don't be afraid to submit something, especially with jobs too. I mean, if you're passionate about it, just try. I didn't do a postdoc, right? That was something you said. I just went right from OHSU to Reed. There was something that opened up. I didn't think I was that great, but I just tried. And it turned out that I ended up getting this job, okay? Yeah. Yeah, some of them else. It's fine, right? You don't need to wait for what other people tell you is the right time to wait, the bottom line. Okay. So zooming out here. So we're going to talk about the green path to purple assembly. So this is just some nice pictures. I didn't get these crystal structures, but it sure is nice looking, isn't it? So I'm going to sort of orient you and we're going to zoom in. So copper A, the protein that I'm going to tell you about, is a small subunit in complex 4 of the electron transport chain. And this is just to kind of give you an idea of where it exists. It's generally in the sort of embedded inside so that we can have a separation of protons and be able to do work. And so if we zoom in here, this is actually beautiful. I think they've actually, since the making of this slide, been able to crystallize this part as well. It's pretty amazing to be able to do that. And essentially what happens there is that the electrons from the food that we ate, we stripped all of the electrons we could from the carbons, and then that finally goes into here, travels through, and it's used to do work and eventually make ATP. Pretty important. And all mammals and some bacteria need this to survive. Any kind of mutation in any of this. So zoom in here. This is where we're going to zoom in. This is cytochrome C oxidase. So like I said, this is cytochrome C. So this is basically the end of it, the end of the road. Mario was talking about this when it came to the terminal electron acceptor in a very simplistic way. 4 electrons, 8 protons, and a dioxygen or element or molecular oxygen will be converted to 4 protons, 2 waters, and then drive ATP synthesis through the proton gradient. So inner membrane space, lipid bilayer, cytoplasm, great. Up here is where we're going to focus. The electrons have to get from one of those subunits to this subunit to get to its final destination. And it uses something called cytochrome C, which is just a little protein that moves it along and brings it right there. So in here in this purple protein, it is actually purple when you put it in a vial. So I'm not just making this up. You can see that there's a lot going on in here. We have some hemes down there. That's where a lot of the business happens. But look how far away this metallocenter in copper A is from this. And in fact, this is known as nature's superconductor, and a lot of work was done by the Solomon Group, Harry Gray. A lot of people are very interested in this, I think, in the 90s and early 2000s. And then I picked it up from there for a different purpose. But essentially what we have here, so I don't know how familiar you are with the amino acids, but you can kind of see a ligand and kind of go from there. So we have a histidine, sort of an imidazole here on one side, a methionine. So this is carbon, sulfur, carbon, a couple more carbons, great. 2 cysteines, so actual sulfurs hanging on there. And then on this side, we have a histidine, and I believe that's a glutamate. So we have 2 very distinct sites that are holding on to these coppers, and they're each in a different oxidation state, which sounds a little bit odd. And in fact, what happens, so these actually, this little bar here is not just for show. They actually share covalency. They're pretty close together. And what this does is delocalize the electrons. And so it's actually a 1.5 oxidation state, just kind of blends across. And what this does is it actually shortens the distance the electron has to travel by about 9 angstroms. So it's actually somehow transporting or making this sort of potential to be able to move the electron a very large distance. I don't know why, but that's why we do the science. The thing I've always been interested in, or what I became interested in when I joined Ninny and Blackburn's lab, is how do we assemble this little subunit? It had to get put through, the usual stuff, transcription, it gets made, but like what is responsible for putting in those 2 coppers? And 2 things can't go in one door at once. And so at least in the sense of not ever having free copper in the cell, there's always a chaperone or somebody responsible for bringing these things in. And so we were curious about investigating this because we actually were able to get a construct from the Abriata group, I think where is Abriata? I want to say Argentina, but I'm not sure. Anyway, got the construct from them and we wanted to start playing around with it from Thermus thermophilus, which is a very heat tolerant bacteria. So for a while there, this is my work from when I was a graduate student, and I'm telling you all of these because they're examples of BioXAS, and so I'm really excited about being able to bring out my best reruns as so to think about it that way. So in the literature, what I knew going in is that there were two possible chaperones that could be implicated in copper A assembly. However, both of these chaperones, so here we have PCUAC, meaning basically a, oh, I can't think of the word, but anyway, it's a chaperone to copper A. It binds a copper 1 in a site that looks kind of similar to half of the copper A. And then there was another protein called SCO. We have some in humans, but it's a little bit different. But bottom line, it shares this kind of homology and looks like this. And it has a copper 1 and copper 2 ability. So it can have both oxidation states. And so the question that that you need to have in the back of your mind, because I'm not going to talk about it much again, is does copper A only need one or both chaperones to mature? It's not that weird that maybe PCUAC comes in twice, or maybe it's one of each. Who knows? Okay, we don't know. But my interest was to start with the empty protein. So up until then, you would purify this protein by basically taking your lysate, adding copper 2, it would assemble the active site, and then you would just literally do size exclusion, purify your protein by the purple band that would appear in your column. And so that was great, but it was already assembled. How do you study it from first principles? Well, surprisingly, most bacteria don't have the excess of metals such that when you purify it without all of that copper, you get the apoprotein empty. Doesn't have anything in it. It's very strange. You would think it would pick up something, but it really doesn't. So this is me in grad school. This is the protein. Isn't it beautiful? It's so beautiful. It's purple. Inorganic chemistry. So this is the mixed valence copper A, 2-to-1 copper to protein. You're going to hear these ratios a lot, so I'm trying to kind of orient you here. But yes, I was very excited the first time that I purified it, and how I purified this was actually from the apoprotein, and then had to reduce the protein to reduce the cysteine residues in a chamber, an anaerobic chamber, and then slowly incorporate the metal. So one day, I was just starting. In fact, I was a technician in this lab for a year because I was first in my family to go to college. Everyone said, what are you going to do with a chemistry degree? Which sounds funny, right? But it's true. It's like, what are you going to do with that? And I'm like, well, I'll show them. I'm going to get a job for money. And so I started in here. And so I just kind of was like, great, I made money for a year. And then I joined the program, of course, because the minute I got on the beamline, I was hooked. So I was taking this empty, it was clear, it was apoprotein, empty protein, and I started, and it was reduced, ready to go, and I started adding copper 2 to it. And I was just playing around because I had buckets of it, just so much of it. And it started to change color, but it wasn't the color I was expecting. It was a beautiful chartreuse green. I don't know the word chartreuse, but it's exactly like a bottle of chartreuse, like here. That's chartreuse. So imagine you're seeing that and you're like, what did I fuck up? Right? You're like, I did something wrong. So I take the vial in to my PI. He's sitting there having, he's Scottish. So, you know, he's like taking his little tea and his biscuit. And he's like, what the fuck is that? So then you do it 10 more times, right? And you do it again and again and again. And it just kept happening. So then, of course, we throw it into a UV vis. That's in the lab. We can check out what's going on electronically. So this is the beautiful spectra of the mixed valence copper A, just so that you have a reference. So there's a lot going on. The purple is actually not like in the purple side of things. It's not, the blue shift is actually red, blue, and a bunch of other things going on, as you can see. But it has this characteristic sort of saddle shape. And then some stuff out here and some stuff there. But what I saw when I started doing that incubation properly, you know, you have your cuvette and you start adding in your stuff and you're stirring, is that with each aliquot of copper 2 that I was adding, I was getting this brand new feature here, along with this over here. And then as I added that, once I got it up to probably about here, the blue peak, it started to collapse. So it started to immediately go into the purple. Something was happening that chemically was interesting. And so we would get from here, so this is just sort of like the time point after, it starts to decrease and becomes the peak that we recognize in blue. Okay, with me so far? Cool. So the question is this some kind of neat thing? And I want to point out that we kept it at 0.5 copper equivalents to 1 protein so that we didn't get that collapse. But then we used stop flow later to see what was going on. So this was to protect the species that we were seeing. It's clearly copper 2 because we can see it in a UV vis. And then we could make samples from there and freeze them. So we have something copper 2. Luckily with this audience, I don't necessarily have to go into the details, nor can I go into the details of EPR. I did once know how to do the hyperfine coupling and splitting, but not anymore. In bioinorganic chemistry, though, if you're not going deep, generally what you do is you look at the Hamiltonian values in the literature and you can compare those to what you have. There's also fitting software, obviously, but you generally can be like, this is kind of like a type 1 or this is kind of like a type 2. So here's what we ended up having to do. This is in collaboration with Pierre Moine-Lacoste, who does a lot of cool stuff, and he and Ninian and another person in the lab, they had bought an EPR and it's still working today. So this is what the copper A looks like, the mixed valence copper A in the EPR. And this was published far before me. Really, really cool. When I froze up some of that green, what you can see is something very, very different, okay? And I was like, great, how do I interpret this? Basically, again, what you do is you go through the literature for these values and you say, what kind of pops up in the bioinorganic literature for proteins that share this? And so what I can tell you with authority is the green intermediates EPR values are consistent with a mononuclear type 2 thiolate center, okay? Cool, great. It's a long time ago. I swear I used to know it. So that was cool. We have the copper 2 in one of the sites somewhere, but how do we know what site it's on? Because this is actually important to us, trying to figure out what chaperone. And there's a lot of work that came before me that studies this basic chemistry of recognition. Lucia Banchi is a person that did a ton of amazing work on this. David Giedrock does a lot of kind of work like this. And so we need to try to figure out, where does the copper 2 live? And if we somehow could just put in a copper 1, because remember, it's a mixed valence, where would it like to live? The answer for me is X-ray absorption spectroscopy, because then I can use both copper 1 and copper 2. I'm putting this up just to kind of like giggle, because this is what I normally will tell people about with XAS. This is my slide that I usually make. And now you can look at this and go, yeah, you can feel so smug and proud of yourselves that this is like simplistic, right? So anyway, what I usually tell people, and I think is useful no matter who you are, is how many of what type of ligands are at what distance from the metal. There's plenty more information you can get from it. Obviously, this workshop has been a total mind blower as far as how many things you can do, but in essence, that's what's going on. And so what I tell people is, depending on what you probe, you're looking at it from the point of view of that metal. So this is actually selenium edge, and it doesn't matter, but here what we're looking at is what selenium sees as it looks across the active site. I think that's useful for people. If you have to explain it to people, your mom, you know, this is a way to do that. Read this for a second while I breathe. The altitude. I'm surprised I'm like breathless. Okay. Plus, I always make my students look at a slide first before I start talking, because you can't write and hear at the same time. Okay, so something that Ninian came up with that I really took off and ran with, and so did he in different ways, is a lot of metalloproteins don't have a lot of sulfur ligands. They're usually in the active site, especially with the sort of borderline metals. You'll see a lot of methionine. And so what we can do is we can force the E. coli in minimal media. You control everything. You grow the E. coli that has your, DNA in there to produce this protein, and you incubate it with selenomethionine, like actually the amino acid. And instead of it having to think about anything or trying to make methionine from scratch, it's lazy. It's like, great, I'll put this in. Okay. And so it does. And they use this for crystallography, but we're using it for a different purpose. The beautiful thing with the row of chalcogens here is that it's really not that far off from oxygen and sulfur. It's just one down. And while there are considerations that we'll talk about to doing this kind of labeling, you know, affecting sort of the distances or affinity or whatever that may be, it's actually pretty nominal, especially for something like this that isn't really a kinetic event per se. So what we did is we're able to then look at the copper edge and see what copper is looking at in this system. So we took that green sample, froze it, took it to the synchrotron, tuned the edge to copper, and then voila, let's see what copper sees. And so when we look at the copper edge, I didn't include the Debye-Wallers. I'm sorry. This crowd would have loved the Debye-Wallers. What? The Debye-Waller factor. Basically a measure of instability or temperature. There's, yeah, basically all the crud, but it tells you a lot actually. But I can tell you that the Debye-Wallers were consistent with these values. But copper was seeing one, it's a histidine, actually, not just a nitrogen. So there was multiple scattering in there, but it wasn't too perfect. And then we see 2 sulfurs. Okay. What copper doesn't see is another copper. So we knew that this green intermediate was just in one side, right? Who knows if maybe it was kind of on both sides. Yeah. And then we saw 0 selenium. So that means, I mean, the distances are just, you know, it's too far. So it didn't see anything from over here. And then when we switch to the selenium edge, basically see 2 carbons, which is good. Yes, exactly. Chef's kiss. And I just want to point out that I did all of the fits for these as a technician. It was great. So if I can do it, you can too. Okay. So 0 copper. All right. Selenium didn't see any copper. And I love this control as well, because you can actually use those 2 carbons in your fits. It should be the same from sample to sample. It's an additional way of getting quality results. So then, okay, we can do this green intermediate. What about copper 1? What happens there? Will that inform us? The hypothesis was, well, maybe it's going to switch over to the other side. Maybe it won't. Maybe it doesn't want to bond. Maybe there's something going on. What I do know is that if you put in 2 copper 2s, there's basically a disproportionation reaction that happens because it really wants to be that mixed valence. So if you just put in the copper 2, it becomes 1 and 2. So that's why we're curious about why would it even have that specificity. So what I did is I made the copper 2 mononuclear species in the glove box, and then I added either dithionite. Actually, I didn't use TSEP because that's a mesh. You could end up with a phosphine ligand. So we ended up using dithionite, which is our one of choice for copper in particular. Used extensive spin columns to get rid of any of that. Since it's anaerobic, you don't see any oxidation, and then took that to the synchrotron. Now, again, you can't see any of this. Copper 1 is silent to any real methods you have in the lab. You can do a BCA, a colorimetric acid or assay, to at least see if it's somewhere in there. But otherwise, you're kind of going without any sight. And so here we go again with the copper and selenium, K-edge XAS of the copper 1 species. Now here what we see is a very different environment. Okay, so we have our histidine, and I'm just showing the primary nitrogen here, but it is multiple scattering. And then we have 2 sulfurs from those cysteines. And look at that, we have 1 selenium at 2.47 angstroms, and a really nice signal. Okay, now the nice thing about this is that the Debye-Waller and the distances should be comparable when you switch over to the copper edge. So it's a really important control in what you're doing. And so then when we switch over the selenium edge, what do you know? It's over there. We've got our 2 carbons. We have 1 sulfur. And then now the selenium sees 1 copper. Did I just equivocate? Well, anyway, this is the copper edge. This is the selenium edge. Selenium sees 2 carbons, 1 sulfur in there, and then, or in there, and then 2.48 angstroms with a very, like almost the exact same Debye-Waller, if I think about 2 or about 0.009, which depending on how you multiply your Debye-Wallers, great. So it's over there. Cool. We can have stable copper 1 and copper 2 intermediates. And from here, what I was able to do is bring it home. So there are plenty of things you can do in conjunction with things at the synchrotron, which is really cool. You don't have to sit and wait around. I mean, it depends on your system. If it's all copper 1 or zinc, I mean, yeah, that's what you're going to have to do. But I basically made an anaerobic stopped flow with a giant bag and argon and another flow thing for inside. So basically you get all of this anaerobic by just flowing gas through. That's the way you can do this in a simple way. And then I had the copper 2 in one of the syringes, and then I had the apoprotein properly reduced and anaerobic on the other side. And then you have a servo drive that basically gives perfect mixing, which in a sort of physics sense, that is a big thing. Fluid dynamics is important here. But what you end up seeing is it starts here, and this is the time course of what's going on. And you saw that in the UV vis, okay? But you didn't really see that like this time course. And what's so cool is these were the kinetic traces we were getting for any of the shoulders. And that's really weird to me. It took us a long time to figure out what was going on, that it wasn't some kind of, because usually you see something that goes down or something that goes up, or maybe it kind of does a little curve, but this is such a sharp moment. And of course, we have a couple of sort of not even realized asbestic points, but close enough. But we have some clean conversions. So what we finally came up with, doing these kinetic predictions, you're really just trying a lot of different things. And this is chemical kinetics. So I think that's important to point out. It's not enzyme kinetics. To me, a little bit easier for this purpose. So what we ended up having to converge and ending up fitting all of this data, so that every one of these kinetic traces made sense at many different wavelengths, was that we have the apoprotein grabbing a copper 2, which led to an early intermediate, which is invisible. Can't see it. And so the I early then goes to a green intermediate. So that's the one that we know and love here. So this is down here is where that early intermediate is. And we really couldn't figure it out because it's just too fast. We chilled it, we did everything we could, but that was it. And then there appears that the only way we could think about it is that there is some sacrificial oxidation of some of the apoprotein, which is actually what happens when you incubate copper 2 with this anyway, is that some of it will be lost and it disproportionates to produce 2 copper 1. From there, the intermediate, and we can't see the copper 1, we could argue all day, but this is what converged. This is what really made some sense. And we know that we can have physical copper 1 and copper 2 intermediates. So it makes some sense. So then once this green intermediate meets a copper 1, we now have the holoprotein. And then there also is the case where that extra copper 1, just sort of from a stoichiometric point of view, could mix with some of this to create the intermediate with copper 1. But from what we can tell from the kinetic traces is that copper 1 seems to be a dead end. If you incubate it with copper 1 first, nothing really happens, okay? So this is the side reaction that we were observing. It's actually really interesting information. You can't go through the door at the same time. So what different chaperones could be doing this? Both copper 2 and copper 1 intermediates are existing. It seems as if copper 2 is binding first and then copper 1. So this is basic chemistry. Who knows exactly if the biochemistry will work out perfectly? And in fact, the take home I have, because I moved on to another project that I'm going to talk about next, is that we haven't ruled out that there are 2 chaperones possible, okay? But the next step was some work by Lucia Bonci, which actually showed that if you incubate 2 equivalents of PCUAC with copper A in the presence of reduced SCO, that it actually plays an oxidative role and may actually create this copper 1 and copper 2 sort of path. But that's as far as we wanted to go because we didn't want to compete. We just wanted to understand it more, like that was the thing. So with that, any questions on this part before I go to the next part? And there's 2 microphones up here.
I have a question. So I was wondering the kinetics that you showed us. Does it change if you believe in the one or?
So what I haven't shown here is that, so the PCUAC is only copper 1. So you wouldn't see anything. When I put, I don't want, I will answer that later. Yeah. I don't want to talk about other people's work without them to talk about it, so.
when you do your exams
Mm.
and you expect copper neighbor
Mhm.
and you see zero copper, yeah, no,
Yeah, that
yes,
guy,
yeah,
yeah.
yes, this one. Where do you expect? Well, I mean, if there was a copper neighbor, where do you expect?
Well, what, 2.4 angstroms away from, or 2.8, I think is the copper distance. But like, say again,
2.8 is very short.
right, it's covalent, yeah. Yeah, the copper A has this diamond core thing. It's really an interesting protein.
Okay, so it's very short.
Yes, so it would be, I see what you're asking. You would see it.
Yeah,
Yeah,
that's my
100%. And I have the data that I don't show here that was already published of what the mixed valence looks like. And so we were able to tell right away, this didn't look like that.
Because sometimes, it's fading at the end of the exams.
This is something, I mean, I do some work on iron sulfur clusters as well. And that was like a fun thing to learn is like, what are you really seeing here? And it's far away, but when you have really clean data with an iron sulfur cluster, it's so gratifying because you can actually see it. But yeah, thank you. That's great. Mario behind you, I think. Out of breath.
The time
Mexico
scale for
City.
the stop flow, what is the? I didn't see the time.
Oh, I'm sorry. You're right. This is from 0 to 2, basically, is the
2 seconds.
yeah, within 2 seconds. And it can be a little hard right here, but we did a bunch of chilled stuff too. But I think we only went as low as 10 degrees. So we Arrhenius the heck out of it, but not too much.
First time.
The first time point, I would want to say 4 milliseconds, but who knows? I mean, if that's like for real, that's what I'm saying. Like, I know that that's what it was, but the thing about the applied photophysics with that photodiode array is if you're chilled, it's for real. It's really cool. And we were able to fit most, I mean, that was the hard part right there because of mixing, but yeah, it was very fast.
Previous slides. In the heat there.
That's the fit, yes.
Still some intensity close to 3.
Mhm.
Is it possible that or is it usually not that?
My simple answer to this was beginner. Kelly, basically. Now there's more that I feel comfortable fitting, but one of the things that I think is very important is don't fit every damn thing. If you're not sure what it is and you're trying to fit the primary stuff and your Debye Wallers are telling you, the Debye Wallers kind of tell you if you want more or less of something. They really do, they speak to you. And so when all of that converged and it matched with the selenium edge, I felt more confident that we could add more, but it was probably, who knows, schmutz. Yeah, some of that apoprotein. Yes, exactly. Yeah, I agree. The invisible one? Yeah, it's freaking fast. I only had, my laptop was locked in here and I meant to read like all my supporting information in my paper. So I will tell you later, I have that information, but I don't have it right now. Anyway, I would love to tell you because yeah, I love that stuff. It's really hard to fit. Yeah. I don't see nothing. Not too basic ever.
It was the complete cell or a purified protein?
Like the cell for the beamline or oh no, I love that question. So no, I don't work on whole cell. I do now a little bit because I'm looking at chalcogen toxicity and nanorods forming. So there's a little bit of that, like the cells turn black. It's kind of cool and gross. But no, we we grow the E. coli, they're happy, they're stirring around. And then you basically tear them apart and you've got this soup And now you have to separate the rice from the beans and everything else. Thermus thermophilus is a very hot bug. It likes to live in very hot environments. And so it's actually very tolerant to purification by a heat shock. So a lot of stuff precipitates out because when you heat it up to like 70 Celsius, it's pretty hot. And then you can go from there. But then of course, with the emergence of histidine tags and affinity tags, this became even more simple. And of course, we get rid of that tag before we put any copper near that because 6-histidines would just eat it right up. But yeah, it's basically very clean, and we use sort of a gel method to see that where the protein is traveling, it's a clean band and we're at the right size and everything like that. So great question. Ready for the next chapter? Yes. No.
How do you synthesize the apoprotein without having any copper in?
The size? Wait,
No, like how did you get the protein without the copper?
well, this is the thing. Surprisingly enough, in E. coli for copper proteins, for some reason, we have not seen a lot of contamination. However, we keep the conditions very simple in terms of what we grow them in. And I don't really have a theory as to why. We never really understood how we got so lucky. But of course, we're using ICP OES, we're using BCA assay, we're of course, scanning apoprotein on the beam to ensure that there's nothing in there. And we don't even have to treat with EDTA or anything. In fact, I never allow EDTA in my laboratory unless we're doing EPR. So yeah, I don't know.
Maybe just quick. So this is not a question, but more of
Oh boy.
has done that.
Oh boy.
No, quite. I just want to highlight that this is just a fantastic experiment. Just this perfect mindset, in my opinion, for XAFs and bioXAFs in the way of like I usually encounter if you have hypotheses like that, like why don't you make a crystal out of that? Like why no, just take it as it is.
Yes.
Make it, take the XAFs and it's just such a chef's kiss set of spectra.
I really appreciate that.
And then I want that also that highlight that. I think this is a take home. There are perfect ways to do experiments.
Yeah.
They're way faster.
And we aren't geniuses in the lab. We're just like, cool, how can we use this? How can we use it? That's the thing. I was a technician, just boop, 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 and I see something green. I don't even know what we were originally going to do, So just being aware, you just move with it and like, okay, you don't need a crystal structure. That's important.
Yeah, I have a question to chemistry. And remember, I'm pretty
This or what slide?
I think what you said is that you exchange one of the sulfur with
Yes.
and yeah, me having studied physics and I don't know what chemistry, I don't know how to do that. My question is, how do you know that you kick out this one sulfur and replace it with selenide at exactly that position? Like how do you control the reaction that it actually does?
This is the wonder of the cell, honestly. First of all, though, it's very important. So normally what we grow bacteria in is literally the matter from animals. It's the byproducts of cows, bovine serum albumin, like it's tryptone, yeast. We're giving them just basically powdered nutrients in this soup, okay? And then what we do is we put in a chemical that looks a lot like a sugar, but it's not one that we have naturally occurring in us. But for the E. coli, it's a very strong signal to start making your protein. We inserted a circular piece of DNA, literally it's circular. It's actually how E. coli reproduce or give each other resistance to antibiotics, by example, is that they're in the hospital, environment, and one bacterium has this really cool trick for resisting penicillin, okay? And it will horizontally transfer that over by basically, it's kind of like bacterial sex, and it transfers it over. But the biggest
innovation, I would say, in molecular biology and biochemistry was the ability to use that to our own design, to make our own circular DNA with the information we want on it, with a code that when you put in that sugar, after they've all grown and there's a ton of them in there, millions of them in there, you put in that sugar and they stop everything else. They don't breed, they don't reproduce, they don't eat, they just make your protein. Okay, so then from there, that's kind of a gross process. It's in this kind of stinky soup and you clean it up. But for the selenomethionine labeling, you have to actually, and this is why it's so cool biochemically, you have to think of the minimum amount of nutrition that it needs. It needs some of the things it can't make itself, but for the most part, E. coli can make almost everything itself. So some sugars, some buffer, very few metals, just magnesium, no zinc, because we can't even use copper. Like usually you'll put iron and stuff in there. And you have this clear solution now. Doesn't grow as well, but what you do is you essentially take healthy E. coli and you slowly build up the population in flask after flask until it gets to the point where you put in this amino acid, the L amino acid, selenomethionine. And you also put in all of the other amino acids that you buy them from Sigma, all 20. And instead of putting in methionine, the regular methionine, you buy specially made selenomethionine. And so it has every other ingredient it needs to make any other protein, but wherever there is a methionine, it will substitute it. And then the thing about the evolution of metalloproteins is that very rarely are you going to see methionines just hanging out for no reason because it will grab onto a metal. And so generally the evolutionary trend has been that methionines and cysteines to a certain extent stay in the active site. And then you can verify the protein to selenium ratio using ICP-OES, which is generally what we did. And it was it gets pretty close, I would say, you know, within error 0.9 to 1. And you don't really see that, but it's there. That's a long story, but I think it's important.
No, I have a question. No, I didn't have a question before. You had a question, but now I want to have a question. So you ran the copper edge and selenium edge both on the same sample, right?
Yes.
So my question would be, what were the conditions and what were the motivation? How hard is selenium edge and copper can you put concentration?
This is why I'm at this workshop. Nobody ever asks me the best stuff. Like, how do you really do this? Couple of things. Okay. So the Blackburn lab, one of the things we take pride in is very, very clean dilute samples. And by dilute, I've gone as low as 75 micromolar in copper, even in iron at times, which is almost suicide. But there's certain ways that we design our cuvettes and the type of tape that we use. We do not use Kapton. We use a type of Mylar film splicing tape. Anybody can use that, but you have to make sure it's thin enough. And then as far as the difficulty of this was actually made, these samples were made in 2012 before the beam got hotter. Do you guys remember? Now it's like 500 milliamps at SSRL, but before it wouldn't automatically photo reduce your samples. We didn't even need to use a shutter. It was fine. Yeah, it was just fine.
Quick, the first time I went to SSRL with our sample, first thing I did was put 4 or 5 sheets.
I'm actually going to steal that idea. I want to talk to you about that because, yeah.
It was Thomas.
Oh, great, Thomas. Thank you.
I have sheets. If I ever want to take them.
I love that because, yeah, it's a real thing. No, we were able to get plenty of scans on it. This was at 9.3, I think. And then for the selenium is a gorgeous scatterer. It is just, there's not a lot of information, at least in our realm, as I think we talked about before, in the pre-edge or the Zanes. There's not a whole lot there to look at in this context. I'm sure there is in other applications, but as far as the oscillations, you can just like see them go on and on, man. Oh my God, this big tape. I'm gonna look so dorky. It's okay. I'm a dork. It's fine. But that is not too hard. Concentrations as far as these, I think I don't like them to go up as high as 2. I don't really believe in doing that unless you are trying to get, like for XES, the work with Mario, we had to get those samples really high concentration just to make sure we were seeing what we were seeing. But for the most part, we keep it 500 micromolar to 1 millimolar max. Yeah, Yeah. And buffers, just kind of typical. But again, we run a blank, really important for us, for the copper particularly. But luckily 7.3 and 9.3, really we're lucky. They're very clean and they're aware of, keeping it clean. Other questions for now? Ready for the next stage? Okay. Oh, copper selenium 2.49, I think. Yeah. Ooh, yeah. 0.48, a little bit off. Yeah. Okay. So now, now that you understand kind of how I use this, now I can go on to the more complicated one. This was actually the second stage of my PhD. It's probably part of, how I really got into the metals community. I was just so excited by this work, what you could do. And I had to give my talk on this right after Serena de Beer. And I thought I'd die because she's my idol. And now we're friends. It's great. So copper and silver weapons in the host pathogen war zone. So the copper A story was very much a fundamental there was an argument in the field, and it was very interesting that I found this intermediate and we just followed it, right? It wasn't like we were expecting anything. Here we had a collaboration with Megan McAvoy and Tiffany Mielman, and they had this interesting system. They were doing some of the stuff more from the molecular biology point of view, but they had found this tripartite, meaning 3 parts, system that could resist copper and silver. But they didn't really have a lot of the spectroscopic stuff. They've done a ton of the microbiological and molecular biological stuff. Like what happens when you knock out one part? What happens when you do this or that? How is it resisting? So then we picked it up from there. So here's the war zone. Okay. I'll narrow it down. So when you eat maybe a bad piece of lettuce, okay? Maybe that happens sometimes. If there's a little bacteria on it, what the bad bacteria, so we have tons of healthy, happy, like 8 pounds of bacteria in our gut. There's some food for thought. It's a big part of us, but you can get one that has basically the ability, it's these different factors that allow it to trick your stomach lining into allowing it into your blood, okay? And when it gets into your blood, that's when you start to not feel so good. But what happens is these giant things called macrophages, and they truly are just like these, they come along and they see, I'm serious. I don't take myself serious, but I'm serious. This macrophage comes along and it just absorbs that bacteria. And when it gets it in there, what it does is it takes it to this little killing cell called a phagolysosome. And what it does, this is wild, it recruits all of these copper pumps and manganese, a bunch of other kinds of pumps, but it just floods this area with metal ions. And not only is it believed that there is Fenton chemistry, so of course you have some radical chemistry, but if you've taken inorganic chemistry, you know the William Irving series, Irving-Williams series. Either way, maybe Williams wants to be first this time. Anyway, anyway, it comes in there and we now know that it will bind to anything useful, especially because bacteria have all of their electron transport stuff in the inside membrane. And so it'll just bind in there and it will kill it that way as well, okay? But for all of that happening, here are all the different bacteria and all the different ways they've found to fight that, okay? And we're not gonna go into Streptococcus, Mycobacterium, Salmonella, all bad things. But we're going to talk about the bad E. coli, the kind you hear about on the news when they take the bags of lettuce and they say, don't eat that because it's got the bad E. coli. The unique thing you can probably see here with the E. coli is this big giant thing right here, okay? So CTR1 allows the copper to come in passively. It cannot resist that. It has to bring it in. Copper is an essential micronutrient. Okay, come on in. But if there's a bunch of it, starts to come in. So then we have some extracellular space, some ligands that come in and try to grab it as quick as it can. And then what you see are all of these different ways that we have, at least for now, shown that first it signals this QR and CUSR, and it makes this giant assembly pump that pumps out the copper as quick as it can. And I'll talk about silver in a second. So I didn't do these beautiful crystal structures, but I put them together because it's a nice way to look at what might be happening during that war zone, okay? When all of those coppers and silver ions are in there. Now, why silver, you might ask? Well, first of all, this thing is only able to bind copper and silver. Any other metal, it won't bind and will not efflux it, okay? So if you put in a bunch of iron, the cell dies if it only has this. The other thing is copper and silver co-mineralize in the environment, okay? And they have kind of similar properties in terms of size, their status. I mean, depending on the oxidation state, their hard, soft, acid, base properties, all of that. And yes, that's all I wanted to say about that. So it is affectionately called the periplasmic vacuum cleaner because it works very efficiently. So what we have are three different parts, okay? We have CusF that's made a ton of. When they see the stress and the copper and silver is coming in from that phagolysosome or whatever, grabs it up as quick as it can. And there's a lot of that being made. Then somehow, before the answers I'm going to tell you, it comes over here to this side. Up here is CusC. It's basically a big barrel that allows the copper to exit. We're not going to talk about it much more, except to say it has a very large barrel, one of the largest barrels. No idea why. Down here is CusA. There's three of them there. It is a proton substrate antiporter. So for every copper, a proton gets moved. And that's how it ends up being able to move these ions up and down. And then here in blue is CusB. So there are 6 of these kind of surrounding this big pump. And it's not your average, what we call a fusion protein. It's not just holding everything together. It actually binds metal, okay? And that is very strange. So there's metal that can come in here. So we think it goes in there, but it also can bind here in this uncrystallized spot. And the reason it's uncrystallized is because it was too mobile for it to be able to, so it can take all these different conformations. And then we knew from experience that CusF and this spot on CusB interact. If one of them is empty, it doesn't have any metal, it will transfer to the other. If the other one has the metal, it will transfer back, okay? So, again, there was this question. Is the relationship between these three the following? And I'm going to make sure you understand this before we move on. So, take a minute. CusB, that weird little spot, that little uncrystallized region in the blue that can bind metal. What is it doing? Number one, very rare for that to be existing. And is the CusF, which is the little sponge that's going around grabbing it as fast as it can, is it transferring over to CusB? And then CusB safely moves it to the pump below it? Because it does have enough space to be able to do that. This was actually an argument that was going on in the community about this. You know, people weren't sure. The other possibility, so this would be silver or copper would travel from F to B to a relay. Or the CusB could be a switch, okay? So in that case, first CusB would have to bind metal. Maybe CusF would give it to it, who knows? And then that would activate something in the CusB to affect some kind of conformational change that would then allow CusF to directly travel to CusA. Why does this matter? Well, again, no free copper in the cell, and it must be accounted for at all times. So you don't just want CusF stealing copper from some important electron transport chain thing. You don't want it doing that. You want it to be very specific. Where are we on time? I have no concept of time right now. Well, let's look, I'll give you the secrets to the proposals. If I don't give it today, I'll give it tomorrow. Okay, so that's the promise.
They have like 20 minutes.
Oh shit. Yeah, we're gonna finish this and then we will start fresh because I actually didn't have enough for tomorrow. So is that okay, guys? Can we do the rest tomorrow? Okay, thank you. Okay, I'd rather give you this story here. Okay. Never gotten the luxury of just telling it. Hope it's not boring. Okay. So there are things you can exploit. This was what I was thinking about when they gave us this system. It's like, what can I exploit here? First of all, very important is that CusF and CusB will only transfer metal to one another if one is in the apo form. That seems kind of trivial, like, of course, if one's full, but there are a lot of examples of things sort of, you know, transferring back and forth. And so then you would end up with a mess. Say if you use copper and silver, some would be in one and the other. The second thing that we can use to our advantage is that the Cus system binds both silver 1 and copper 1, okay? Silver one is a logistical nightmare. I will tell you about it. It's the same thing that's used in photographs. So have to make it in the dark. Third, cus F and cus B can both be selenomethionine labeled. So let's go into the zoom of what these look like. So here are the active sites essentially of, and we don't really have an active site per se for the pump, but here's what we have. Don't look at the experimental design yet. I'm just going to show you the various amino acids here. So cus F, when it's bound with copper 1, for example, it's in here. We have a tryptophan that is like a little cap. And then we have 2 methionines. And then we have thank you. You in the back. Here, what we have is cus B, the N-terminal region. Now, it's important for me to tell you that if you cut that off of the main cus B and then produce the DNA separately for that little fragment, the cell will die if you only have the big guy. But once you actually have it overproduce that little fragment again, we restore activity. So this little fragment actually is really determining what's going on. So that was really important because we're not just like taking one little bit. And then the cus A, it's somewhere in here, but it's hard to say. But here's the deal. This has 3 methionines. How lucky could we get? Okay. That's how it binds. And in fact, it leads to like kind of dirty excess because there's not a lot going on in there, which is an interesting thing. So we're going to test the relay model and the switch model by using tri-element biological XAS. Okay, this is what we were talking about, that you can do various things with the same sample. Okay, of course, there were many samples, many controls, but we'll go from there. So experiment number 1, I made this construct with the selenomethionine. I put it in the dark and I very carefully loaded in silver 1, kept it in the dark on ice as long as I needed to. This took me a long time to do, and I had to like keep the ice every day in my glove box. So once that was safe and sound, whew, but then I had to make the kind that didn't have the selenomethionine. Then we have the cus F with copper 1 here, and then here I made the selenomethionine version. Okay, both of these had copper, both of these had silver. So there's 2 sets of experiments we can do. If cus B is the relay, then when I put all 3 of these together, oh, and by the way, I had to make the membrane protein too, and that actually really limited our concentration because membrane proteins, when you try to concentrate them, they won't concentrate. So I had to think very carefully about dilution and how dilute I could go. And this was the one that was about 75 micromolar. So if it's a relay, when I put all 3 of these together, you should see the silver disappear from the selenium signal and copper inhabit this signal here. If instead it's a switch, what we might see is that because cus B already has its silver, it should be ready to go, should be able to relay, or I mean to switch. And then we would see the copper signal disappear from cus F, ostensibly to go to cus A. But what's great is that we can check at any step whether any of these have moved. Does this make any sense? Try doing this in 5 minutes at a data blitz. That was a lot of fun. So I have many more Fourier transforms that I'm not going to show. If you're interested, I would be happy to show you all of the controls that I did, okay? But this is the first experiment. So now what we're checking is if cus B is a relay. The first thing that I did is look at the selenium edge. That's the fastest place to go to start. And so what we see is the selenium carbon. Awesome, all made sense. And then what we see is a strong selenium silver. I don't know why, and maybe the physicists do, but I always get this double peak for selenium silver. It's part of the same waveform. It's not like a second shell. It's so interesting. Anyway, silver is a weird thing to be working with anyway. So we basically saw nothing happen there. And then when I incubated, so this is just cus B and cus A, these 2 together. That can sit there. And then when I add the cus F, we activate, okay? So then what happened when I added the cus F is we saw a little bit of a decrease, about 30%, but as was pointed out, the basically the occupation or occupancy is a little bit wiggly, so we don't want to put too much stock in that, but we saw a change of some kind and a dampening overall because we have more things in the system. So we saw just a slight change here. So this really didn't help our hypothesis that maybe it was some kind of a relay. Nothing really happened, and the copper didn't inhabit this. And of course, we can do the copper edge. We can switch to copper edge and see that it's in a normal CUSF. We had already done that and seen that it hadn't really moved. But we also have this side of things. So then this is where we have the CUSF labeled with the copper. This is the other kind of side of things. And now what you can see is if we have a relay, again, you would probably see the copper disappear and maybe it would go over here and then over there. That did not happen. And before you ask, the selenium copper and selenium silver are mobile. We have shown that if you take CUSF and CUSB, they will readily give it to one another, even if it's labeled, okay? So it's not a dead end. So here's the selenomethionine CUSF with the CUSA, so these two together, and then this is going to activate everything. And so you can see this beautiful selenium copper signal. And then when we put all three together, I about crapped my pants. And yes, we did it more than once on different system or different preps. We see that the copper silver is completely, or copper selenium is completely gone. And now it is inhabited by selenium silver, okay? So what we have is somehow we are causing CUSF to release metal. And consequently, it's taking on some silver. Does that make sense? Okay, I've seen kind of a medium that makes sense. I'm used to seeing that in my classroom, so. Remember how I told you CUSF and CUSB will transfer metal to one another if one of them is APO. So what happened is that, oh God, my breath. Oh, I just wanted to show this. I have a thing that's just, I just want to show I got silver edge. I did simulate, but this is a comparison of two. This is not the fit. This is the before and after. But what I wanted to point out was the weirdness of the phase thing that we had talked about a little bit with Mercury, was it? Or yeah, lead, thank you. But look at how the oscillations don't even really get partying until about 6. It's really interesting. I wish we could have taken it further because I think silver's what, yeah, that was fun. We had to completely detune. We had to fuck with the voltage. It was really fun. Anyway, back to this. So if one is empty, one will transfer over. So what happened was that we activated everything with CUSB. CUSF then was free to drop off its cargo, and then it was empty. And what happens when these guys, one of them is empty? It back transfers some silver, okay? That is what happened. And I say that because I did edges of everything and every control. If you switch to the copper edge on that sample, we saw a completely different environment, okay? So that was after the reaction. All of the copper went somewhere. And CUSF delivered all that copper somewhere. We had no idea where. And it took on some silver from CUSB. When we went to the copper edge of that same sample, which I did in replicate, you see now that we have a non-multiple scattering copper oxygen / nitrogen, 2 copper sulfurs, and this nice little low intensity excess. Now that to me fit to the distance was consistent with 2 methionines. And then I did see some glutamate. And then the crystal structure, what was posited by the people who did a beautiful crystal work on this giant protein, posited that the entry site was actually this right here. And so I put the connections here where what I was seeing was happening. So I didn't see that 3rd methionine, but it was there. And then we had the glutamate. So this actually really fit with what was posited from the crystallography. And the crystallography is less sharp. A lot of the error from crystallography kind of gets put into the metal site. And sometimes the metal isn't there. So this was really nice to see that it was consistent. Now, you know, you can wonder, maybe this isn't in CUS-A, but it was consistent and the stoichiometry was there. So this is the end. These results tell us something very interesting. So my hypothesis going forward was that if the copper concentration is high, we're just thinking about physiologically, like what's happening in the cell when this is happening. If concentration of copper or silver is high, panic, 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 we make this big giant thing. This gets made in excess, it starts grabbing as much as it can. And then it loads cusp-B, because whenever cusp-B is empty, somehow a cusp-F finds it, and vice versa. Then once the cusp-B is activated, somehow that activates the cusp-A pump. We'll talk about why in a minute. So then once cusp-B is ready to go and loaded with metal, then cusp-F is free to deliver its cargo, okay? Why would it be important though, that when copper concentration is low, cusp-F will exhaust its supply as what we saw, and then it will begin to take copper back from cusp-B, and sort of in a smooth gradient that is very in keeping with how the cell is being shown to work in more fluorescent studies, more of like a equilibrium process, like it's a whole process of things. You want to be able to turn this off, because if cusp-F has such a high affinity for copper 1, which is the primary oxidation state of copper in the cell, you don't want it to just be on all the time. And actually, fluorescence group did this amazing work to show that this doesn't just get made when there's shock. It's actually around all the time, but kind of apart. And then once there's copper stress, it assembles together. And so it is really important that this doesn't just work all the time. So it's important that it starts and it stops. And so with that, I'll have you take a picture of this, just because I have a lot of other stories of cool BioXF stuff you can do. The one I'm particularly proud of, actually, like truly, is a collaboration with Mario, actually, because it was I would say it's one of the first ever, if not the first, XES on actual proteins that found copper. So we used PIM, we used cusp-F, we used cusp-B, we did a selenomethionine labeled thing. And I didn't know anything about what Mario does. It's very, like, I was like, you guys do the, the calculations, and you did. And it was really freaking cool. So I'm very proud of that, that you can work together. That's what this field is. Very collaborative. You don't have to feel stupid because you can't do it. Get on the beamline, see what you can get. There's bound to be somebody that can look at it and help you figure out what it is. So that's that. And I'm not going to talk about this right now. You'll have to wait. Thank you. How late was I on that timing?
You're great. You're great. Yeah. Are you going to talk about the intestinal mucin? So you go back.
So yeah, so
Could
my
you
last
tell us
two
about
papers
this
were
tomorrow?
collaborative. Yes. That's my itself. It's so exciting. I will be happy to talk with you about that. I figured since it wasn't directly my discovery, I didn't want to present exactly on it.
It's just that this is so.
I know, I'll talk about it with you offline. Yeah. It was really cool. They study mucin in the gut and they're like, what is this copper doing in here? And me and Kathy Franz said, we can figure that out. It's very fun. Oh my God. It's just finally, we didn't know how the copper got in. So that's really, really cool. Other questions on stuff?
First of all, thank you very much. Beautiful explanation. And my question is, in the process, what does cusp-C do? Because,
That's a great question.
because the copper goes, if I understand correctly, the copper goes to cusp-A, that is at the bottom, and
How does it get up there? So this is something I didn't have time to talk about, but it's in the paper, so you can always look at that. Here's a couple of things. Number one, these channels, this one in particular, is extremely hydrated. So, and it also has a bunch of, what are the amino acid? Yeah, there we go. Tyrosines in there, all kinds of, but mostly acidic residues that hold on to water. And like I said, it's one of the largest channels. So this is C here. Here's, we'll call this B, and we'll call this A. Okay. So first of all, as the copper one is traveling through, I'll explain in a minute, but it's increasingly getting more oxidizing as it goes up. Because one of the questions someone asked, it was really a question, is why, when the copper one comes out, does it not just go right back in again? But ostensibly what we saw from the sort of oxidation state of everything, or like the oxidative amino acids that are in here, is that by the time the copper 1 makes it up, it's generally oxidized by that time. And copper 2 is much less dangerous to the cell. The other thing is, how does it get up there? Did you notice those methionines right there? That's the entry site. There's something called a methionine wire. And what it is basically methionine. The reason why it occurs in metalloproteins so often is because you can hold the metal close enough, but still allow it to move along to the next destination. And so if you have your little sulfurs here, and remember these have methyls on them, so it kind of softens them a little bit, is what I would say. So you can basically decrease the distance of those methionines as it travels upward. And I don't have the crystal structure details here, but there are methionines literally perfectly lining a little channel, and it just teases it up. And basically by the end, it goes into that water-filled channel, and what we hypothesize is that then it pops out oxidized.
Thank you. And the second question is, you said that not any metal can be, because
Just silver and copper 1.
neither gold 1.
Oh, yeah, no. Believe me, it was an expensive experiment, but I did it myself. I did not bond. But yeah, Megan McAvoy and co-work, we also did a lot of ITC, so isothermal calorimetry, to also look at binding and see, and like nothing else but those two. It's something special. That's what's so cool about the fundamental chemistry. Like, that's what excites me. The proteins are awesome. They do cool things, but like, how?
Thank you.
Thank you. Does exist a simulation of the transport of the atom between the subunits?
Oh, that's a great question. I mean, I don't think anyone has gone back to do any work on this, on that uncrystallized part. So this part right here. If we could get that to crystallize, then yes. However, with new things like AlphaFold, I'd be really curious to know if you could at least simulate that, but none exists now.
And this simulation should be classical or quantum or coarse grain?
That is not a question for me.
For Niels.
That's why we're here as a family.
Covalency is involved. I would not go.
Oh, thank you for the great talk. Did you do sulfur etch of these systems?
Not yet. Now, the thing is, no, I should. There's no cysteines anywhere else that would get in the way, and no other methionines. You know what? We did do it. As a proof of concept, I do have that data. In fact, I will show it to you when we don't record tomorrow. Oh, no, we'll wait, we'll come to you.
Well, my question, when you're talking, I mean, the history of the assembly of this particular site, I was thinking, I mean, at the beginning of the generation of the cluster, whenever this protein is assembled. So have you ever thought to yourself, like, if the complete assembly of this protein happens in the same way that you're like proposing here? Because I suppose that at the beginning of the formation of this assembly, there must be changes in the
just sort of,
Copper in the copper necessity for the cell.
yes.
The other is-
So one of the things that's important about this system is that it, and then this may not answer your question, but just sort of obliquely, is that this is only necessary completely for to survive under anaerobic conditions. When you're under aerobic conditions, there are multi-copper oxidases that can safely take copper to and then downstream produce energy or do whatever it's going to do with it. When it's anaerobic, which actually the E. coli will find itself in that situation quite often, that's when this is really, really important because there's no disproportionation happening anywhere else. Does that kind of help? Yeah.
I like technical questions.
Yes.
So did you,
So excited.
so selenium is it like 12?
Yeah,
Copper is it like
12.3.
20?
Mhm.
Silver is it
23 maybe?
3? So how hard is it to go from one to the other?
I found it to be, I find it, we do a lot of edge switching. I enjoy it. I always mess something up small and then have to figure out what happened. You know, there's always a little something. Now, for the most part, I can get started on the beamline 2 hours or 3 hours after I get started. As far as what we do is I try to prepare and at least have my detector windows, like you just kind of keep things. So you never get rid of a file. Most importantly too, is to not get rid of your regions files. So really, we're really picky about, I'm picky about where I, you know, how long I collect for my, you know, pre-edge and how long I collect for the actual white line. And then of course, going out, I'm pretty proud of trying to go out as far into K space as I can, because that's where you can get the most. So with that said, you can go out to copper, let's see, how far out did I go? So we have a couple of different settings for copper depending on what we have in the system. So this is selenium edge, copper. Oh, I only went out to 12. That was, generally 12 is fine, but I've gone out a little bit further. Once you get out further and further into K space, you might as well go have dinner because it takes a really, really long time, but that's where you're collecting the most important data. So then when you move over to the selenium, you can actually get up to about a K of 15. And that means you're there scanning for quite some time. And then silver, as you saw, unfortunately, that was I mean, nobody had really done it in a bioXAS capacity. So we were really just doing it all by hand. I was. And again, this is not a fit. For those of you in this is two things superimposed. We do have fits of it. Is that we went out to a K of 14 and I think we should have gone a little bit higher. I wonder though if there's anything that would get in the way. I don't think there is actually. Does anyone have their data booklet? No, that's an inside joke. Okay. So it was, and then switching to selenium, it used to be a little more difficult, but now they've completely taken the mirrors out of 7.3. 9.3 though is a different story. And when we're doing all of that, we have to be really careful with how we're doing things so we don't fry anything out. But for the most part, it's pretty smooth. We're used to it.
So the noise is due to a reduction in photon flux.
I think so. Yeah. I mean, this is something I want to play with more, but who has any silver? I guess I could just make a bunch of this and play with it. That'd be really fun.
So there you have silver signal post reaction 70%, 30%.
It roughly matched what we had from the selenium silver data.
But it's only taking the exons, or is it also taking the edges?
Oh yeah, we've got all the edges for this. Yeah.
But is it also included in the
Oh,
the
good
edges.
question.
As you will see in my talk, if you take both of them, they can also be consistent.
I like that idea. In fact, it would be nice because it seems like where I'm starting E0 is all messed up. I couldn't quite get it. And this was me as a grad student, just like, what am I doing? Playing around with it. Nobody else wants to touch it. So that's the other thing as a grad student, play with the things nobody else wants to touch. Seriously, it's very fun. You have nothing to lose as long as you have your main project. Yeah, good question. I would like to play with that a little bit. Still have the data.
It's the last question.
Thank you. I also have another technical question. Since you said that you know, it takes very long when you go to K values, when you go for dinner. How do you like as a general explanation. How do you, or advise, how do you deal with like X-ray radiation damage? How do you check if you have some, if you don't have some? How do you make sure that that your data is actually the actual protein or enzyme?
Yeah.
How to
Well, first of all, we operate at about 9K. So we go as cold as we can. The other thing is that our cells are extremely thin. So one of the things that really helps with back fluorescence transfer or any kind of effects where you're having internal absorption with these tiny little cuvettes, it's only, what? I will tell you later. It's not my information to give, right? You inherit things from your PI and you, yeah, you're like, but very thin. And like I said, we use almost the thinness of tape you would use at 4-3, like that stuff that doesn't even stick, film splicing tape, which is like Kapton. And then as far as that's concerned, now that the beam is hotter, we have a huge amount of troubleshooting to do. Basically now the trick is you bring multiple samples and you take multiple spots. You compare your pre-edges, you see how it's looking. For the most part with selenium or any of these really robust scatterers, you don't have as much of a problem. Oddly enough, everything's kind of frozen in place and I don't usually see any kind of damage. However, you could never thaw out the sample now. It used to be I could possibly thaw out one of these samples and use it for something, but now it's completely obliterated. So it's almost like LCLS with the mini crystals, right? It's like one shot, it's broken, it's done. But we get it. And we now just change spots with the queuing system, which thank God they came up with the queuing system so that we could go to dinner. Yeah.
that's another question. You said that
The copper, this one was like my proudest moment here. Yeah,
that's quite a
of course, but it's also because there's no multiple scattering. And it's two methionines, not two cysteines. So the distances. Yeah, true. The cryostat. Yeah. You think? I don't know. I did the blank. I didn't see anything. Oh, at Bessie. I was like, are you saying this is the cryostat? The reviewers would have said that's the cryostat. So yeah, no, thank you. Yeah, exactly. That's why, I mean, I've gone to many different beam lengths. They all have their strengths. SSRL's strength is that they actually think about, not that Bessie doesn't, but it took them many years to make a setup where when you run a blank, you don't see a bunch of crap. One more, or are we done?
Yeah, I mean, you're raising a fantastic point. I mean that, but on the other hand, I think.
Oh, I don't mean you're done. I mean, should I get off the stage?
Yeah, we should get dinner. Like at Bessie, I can run zinc a lot better than SSRL, but it's SSRL, I can run copper a lot better than
Copper.
But the problem is I have copper and zinc samples, so I either like which one should win this time is the question.
Completely agree. Yeah,
And-
zinc is the glitches on zinc are just so hard at SSRL.
And now the actual question. You mentioned, and I want to follow up on that, you don't use chemical. Why? Yeah, like, is there a very obvious?
It used to
explanation?
matter more. Here's what I'll say. It used to matter more. And when you do something a certain way, it's just like when you were talking about fitting, be consistent. Who knows? It's like, it's only weird if it doesn't work. You know, it's only superstitious if it doesn't work. So, you know, for me, I think the biggest thing is the cell size and the path of that. But then the capstone or the Mylar, it's really nice to know it's not doing anything. The other thing though, if you're on 4.3 is that, yeah, we can go into that another time. I just do it without, I do a cryostream and no vacuum and I just have nothing on it. I had to make my own little matchstick samples for that. Okay.
Okay. Do we know in which subunit the oxidation from copper 1 to copper 2 occurs?
I was
Okay.
very curious to do that work, but I got a job.
Okay.
I ended up getting
Yeah.
a job and then you're like, see you later. It was great. It was great. Okay, I'm gonna stop now. Thank you so much, guys.
Yeah, I think very warm set of applause for it. Okay. So now we are switching to the last hands-on session. Then you're officially schooled in synchrotron-based X-ray spectroscopy.
Quick thing, I just got out of the Zoom. Is that appropriate?
Yeah,
Okay.
I think so. We're not recording the last session.
Oh, yeah, that's
try
right.
to
We're done.
share
Yeah.
All the secrets of how to solve such as. Goodbye. Thank you for tuning in. The one adult streaming site that is posting. I don't know if you've seen chat.